whatever research you're pursuing, whether you're studying or you're, in a job, or you're training, you're training others or you're teaching others, it's extremely important that you understand data science entirely. And in the next 50 years, biology would be summed up, if biology could be summed up into one word, it would be integration. Already, more of you are spending more time on the computer, and the internet to generate and describe your data in experiments. But recently, and because of the pandemic, this entire year, I think you've spent far more time on the computer, even to communicate and for basic stuff like, even health and household grocery shopping. Everybody's been on the internet, and therefore, including research, all the more so in research, you would be expected to analyze your data and find other people's data relevant for comparison, and that cannot be done without the help of the machines or the internet. And therefore, in order for you to understand, find new knowledge, and explore existing knowledge in the field and relate it to your own results, finally into the current body of knowledge to publish to the world, you would need to understand data science and computing as best as you can. I am a huge proponent of R. Today, of course, much of the effort is about computational biology networks. And so I will be focusing on, other than R, Cytoscape also, but the power of R for data scales is absolutely unbridled. If this is a successful, if this is a successful, let's say, meeting or workshop today, next year for InCoB, I would like to offer a longer course on our data skills with R. We already are offering several such courses at the University of Cambridge, where I'm an instructor in bioinformatics training. You're all welcome to visit the Cambridge University training website, because you will get to see the diverse kinds of trainings that we conduct over there. And here is a small list of the kinds of training courses that we've been offering at Cambridge. Some of them are core and specialized core skills, and others are specialized skills. Among the core skills, we are teaching Introduction to Python, Object-Oriented Python, and Perl programming. And among the core R skills, which is my particular forte, we are teaching Introduction to R, Advanced, Intermediate, and Core R, Tidyverse, ggplot, and dplyr. And this course, basically, these are not 1 hour, 2 hour, or 4 hour courses like we're having today. These are courses ranging from 2 days to 4 days, where we give you online workstations or remote online workstations, and then we give you the data sets and the pre-installed software beforehand, and then we type out with you. So we do interactive one-on-one training, and these trainings can obviously not have a lot of people, so that it has to have a limited number of people so that we can reach out to most, so that you gain the most out of it. And the next course in this series is being held on 14th to 17th December. I welcome all of you, if you're interested, to participate in this one. This is a course, all right, but this is only for women. So this is an effort towards women in science. So it's an introduction to data science for women, and it's a 4-day course that I'm organizing. You're more than welcome to, if you're a lady scientist or a girl student, you're more than welcome to apply for it at the link given in the booklet. I'm going to share the course booklet with you in the chat box, or I may request Sita to do that, so that you can see more about it later on. But this is something that is happening next week. And so, okay. All right, so it seems that I'm being asked to make full screen. Thank you, Shofi. I would, in that case, what I was thinking was, because I'm sharing these multiple screens at different times, I'm using the online Chrome for this workshop. What I'll do is, once the introductions are over, I think I'll move on to PowerPoint, and then we can have a full screen. So I hope that's all right for everybody. This is still the introduction. We're not really going on to things. So during the COVID pandemic, what I did was that I offered a lot of protocols from home. And during this, I tried to train students, anybody who might be interested in the world. I made these public and free, and I was just teaching from my home on various topics. I'm still continuing to do some of these, but not as often, because we have got back to work now, and there's a lot of things we're doing. With AP Bionet, we're planning more tutorials. In case any of you is interested, Please join me on Twitter. My Twitter ID is JillianV, and you can find new courses that we are offering there and updates about these courses also. Among the biological network courses, there are 3 basic courses that I offer, of which the first one is being done today. This is Basic Biological Networks, where you will understand why it is important to visualize your data as a network. You could be looking at genomic data, you could be looking at tabular data, but you can always convert it to a network, and it's not difficult at all. So that's the ice breaking we're going to do today. You will also learn how to visualize your data as a network, and you will learn several other layout algorithms. There will be a hands-on tutorial. I had initially thought there won't be enough time, but with C2's help, I've been able to come up with a short stepwise tutorial, and I really request you to, be part of that. So during the break, which we will have, I will give you instructions to download and install the Cytoscape, software on your laptop. You don't have to do it now, so you can keep listening to me, and there is no need to install or do anything in the first part of the workshop. You will get a 30-minute break. During that break, you can do some installations, optional, that is also not necessary. And after that, we will have a stepwise session for those of you who might want to really dip your hands and fingers into the networks yourself and try out what happens. I will also give you some tips for effective visualization of networks, and we will talk about network clustering, module detection. Basically, it's called community detection in biology, and so on. And finally, if we have time, we will do network motifs. I have not originally included network motifs in this in the first course, but today I will do it because I think this is my first InCoB workshop, and I do want to be able to leave you with an idea that it's good to use networks and to work with networks in your own data. So we will also look at transcriptional, my other courses are obviously transcriptional regulatory networks, where we do construction and analysis of unweighted gene expression networks, or gene co-expression weighted networks like the WGCN analysis and so on. And there we detect consensus modules and eigen gene networks and everything. So that's another set of tutorials altogether, which is an advanced course beyond the one we are taking today. If those of you who are interested are more than welcome to give us a feedback that you're interested in getting the other course done. Then maybe next year I'll offer that one instead of the R course. Let's see what kind of feedback we get. And then, of course, the third one is a bibliography network course, but my students are much better at doing that now. I'm realizing that students can often go far beyond what your own expertise is. Today's session info is that we can not have a large number of audience. So I had requested AB Bionet to keep a max of 50, but I can see very clearly that we are well under 50. And therefore, I'm very happy to realize that we can have a more one-on-one interaction. And this is mainly for students and early career researchers, but we will have an introduction from you where I'll try to get to understand what you are doing currently in life. There are no prerequisites to this course. This is the most basic biological network course that we take at Cambridge, and you essentially need nothing. But I will ask you to download Cytoscape in the break to make you try some things out yourself. The aim of this tutorial is basically just to give you an introductory experience of network concepts, and also how to use networks, create and visualize networks in R. R is the global open source programming language, which I talked about earlier also. Earlier, I told you R is the best for data skills, but R is the best for everything, absolutely everything under the sun. And so you must, if you haven't learned R, pick up R, learn R if you can, and join us on our December workshop for 4 days if you'd like to really get a step-by-step introduction to our working environment, as well as data manipulation and visualization in R. You may want to, I'm sure that everybody who's online is listening to this tutorial on their laptop or workstations or desktops. You might want to, after the break, download Cytoscape or R Studio. Cytoscape more importantly, because the R stepwise tutorial that I have, I'm not expecting you to be typing that out. It will need more time. With R, even if it's a short course, it needs more time. So we usually go into days. I would much prefer if you have Chrome or Edge browsers installed on your machine, but in case you don't worry about it. The timetable for today's session is that the first 10 minutes will be for this quick overview and introduction with each other. I can see that I'm already touching the end of that. And so this will be one of my second last slides, I think. And following this, I've not put exact time that this is 1 p.m. Singapore time or 10:30 a.m. here in India where I am, but basically I've put it in slots of time, whatever time, wherever in the world you are, you should be able to know that the first 10 minutes of this workshop will be just an overview and icebreak introductions. And then we will have network concepts, representations, topologies, creating and visualizing networks in R as well as in Cytoscape, and then clustering, followed by aesthetics, tips and tricks. Then we'll have a break. In the break, I will ask you to, install the free software. It's a free software. You can use it on any operating system. And then we will have, after the break, we'll come back and do things manually. You will set up your workspace. You will import a publicly available network, because in the first part of my talk today, I will already tell you how to create a network from scratch. And so therefore, in the later part, I will teach you how to import a public network, how to explore it, and then how to apply different styles for visualization and layouts to it. There will be an exercise and a small test. Let's see if you can, some of you can do it. It's not meant for scoring you, but just for you to understand how to do these things. And then, of course, I'll teach you how to cluster and annotate your network. Finally, we will learn how to save a workspace so that you don't have to repeat everything. Reproducibility in science is the foremost important thing, whatever you're doing, whether it's experimental work, computational work. Reproducibility in research is more important than anything else. And I can't emphasize it enough. You've got to know how to reproduce your work. Your peers, your reviewers, your examiners have to know how you reproduce your work. And so therefore, those codes and in Cytoscape, the sessions are usually available for others to share, for you to share with others so that they can reproduce your work. Even you coming back to your work after 4 months, 2 months, even shorter time sometimes, you can't remember what you did. And so therefore, reproducibility becomes even more important, not just for anybody else, but for yourself. So with that, let's go on to the next slide. This is the introductions and ice break. I would like all of you, I want to get to know you first. So what I would like you to do is go to this website called www.menti.com and use the code 3320700. I request everyone to please go to the website, open a new tab, go to the website www.menti.com and use the code 332070. Okay, and let's see what happens. Has anyone tried it yet? So there are questions there. The first question is, which country are you in? And here are the answers. I think everyone can see, and that is why I was using this slide. So there's people from Malaysia, people from Indonesia. Okay, there are some people from China who forgot to type it correctly. And of course, there's a few students from India. I expected that also. Very nice. All right, we have more students from India. Oh, there's someone from France. Very interesting. Okay. All right. Has everybody answered this question? Indonesia, France. All right. There's somebody from the USA. Oh, it would be too early for you in the USA, or too late. Would it be? I think it would be too late for you. Okay. Very nice. In France, it would be too early. I welcome whoever is here from France. You've really woken up very early in the morning to be here with us. Thank you so much. Lovely. All right. So that's your first question. Okay. I still don't have enough inputs. No, not everybody has logged in. I can see that we have more than 20 participants here, but only 12 have given an answer. Those of you who have not logged into Mentimeter yet, please go to www.menti.com and type in the code that I gave you, which is 3320700. That is 3320700. Oh. Okay, I have some smaller answers elsewhere, but I can't see those very clearly. All right, so this does give me an idea that you're mainly from India, Malaysia, Indonesia, and we have very few students from other parts of the world, and that's very nice. How about the next question? What stage of career are you at? This will give me an idea about whether or not you're studying currently, or if you're doing research. I mean, if you're working as a postdoc, or if you're yourself teaching, or if you've got something else altogether which I couldn't come up with. Let's hear. Okay, we have some students. Okay. All right, we have some postdocs here also. Very nice. I still think that not everybody has yet answered the questions that we have. And so in case okay. Wow, it's 6 a.m. in France. Amazing, Yasmin. Welcome, very welcome, big welcome to you. And once again, please see the chat box for the menti.com code in case you haven't seen it already. The code is 332070. That'll give me an idea about each one of you. And I especially encourage all of you to please respond to this, because that's the only way for me to find out about you without putting a lot of time there. So, all right. So the main thing is that among us, I think we have more students. We have a few postdocs, and that's very nice. So let's get right back to trying to get to understand, get to know you again. Now I've got an idea about who you are. Now I want to know about your work. So can you please go to menti.com again and use this time another code? The code is called 7330743. Shofi, I'll be grateful if you can type it out in the chat box. You have to go to menti.com and use the code 7330743. With this, you'll have a question like, what's your species of interest? Because quite often it helps to understand whether you're doing biomedical research, plant research, are you a zoologist, or are you looking at chemistry altogether? And so not at species. It lets me get an idea about what you're doing. So, okay. All right, species of interest. And okay, the first question is about, it's about whether you've used R. Those of you who have used R, there are those of you who have used R only. There's somebody who has used Cytoscape only, and there's somebody who's not used any of the two. Okay, so I'm very happy that there is someone who's never used any of these. So that will help me. Oh, good. So there are more people who've never used any of the two. That will help me to understand the extent to which that will help me to know that I'm going to be reaching out to people who've never seen any of these two fantastic softwares, and I do hope that you feel encouraged to use more of them in the future. So most people have used R, great, which means Cytoscape is new for you, which also makes me feel very good that you've done that. This is going to be a useful exercise and tutorial for you. And the next question that we have is about, I think, the species of interest that you have. Okay, we have people who are working with Homo sapiens. Okay, you've put your area of interest. That's all right. That's also a good way of, that should have been my question, is to the species. What's the area of interest that you have? Biology, bioprocess integration. If anyone is working on any specific microalgae, very interesting. Those of you who work on cancer, oh, very nice. So there are cancer researchers here. Who else is there among our students? Okay, microalgae, humans. I think everybody has not yet answered. What you can do is to go to menti.com and type the code 7330743. If you don't remember the code, you can just go to the chat box and you can answer it from there. So 7330743 and 3. Okay, Mycobacterium tuberculosis, very nice. There's someone who's interested in systems biology. Okay. Oh, now I have more answers. That's all very nice. So in all of these areas, systems biology, complex networks, well, bioprocess integration is new for me. I think that in all of these these research areas that you're working with and the species that you're working with, you would have collected some kind of data. That kind of data would usually be in the form of either surveys or tabular data, like with rows and columns. The rows and columns might be genes and their information. The rows and columns might be species and something about them, or experiments that you've done and the results that came out of them. What we will see today is on a large scale how to convert these kinds of information or data sets into networks. Those of you who have specific questions about your particular area of research and how you should try and convert it to some kind of, how you could convert it to a network, you're more than welcome to ask questions on the Discord channel or in the chat box here. I'll be more than happy. Discord channel is probably better because the chat box will, as Shofi had mentioned at the outset, the chat box will disappear as soon as the workshop is over. I will be following the chat box during the break. I will take that time to respond to each one of your questions, but to have a long-term interaction and to keep a sustained interaction during the conference, I think it's extremely important that you put your questions or copy your questions into the Discord box also. And there, C2 and I are both available to be responding to the different questions that you have. And I would love for you to have specific questions about the kind of data you're looking at and how to have ideas to convert it. For example, I have a lot of friends who are ecologists, and they collect information like species abundance patterns, they make transects in various fields, and more often than not, they were doing ordination and statistical research. So when I met a student like that, one of the students had the question about how this can be converted to a network. And it was so simple. We built a unipartite and then a bipartite network. We will talk about those networks today, but I'm just giving you a small snippet of an example whereby building bipartite networks between the spatial locations and the species or species of trees and animals that they had in those spatial, that they had found in those spatial locations during their ecological research surveys, we were able to do community detection in such interesting ways that we were able to find what kind of species associations occur more often and how those species associations are connected to where you are, essentially. So there's a huge environmental factor playing a role there. Similarly, if you have protein-protein interaction data or gene expression data, or even if you don't have that data but are interested in that data for your vaccine target discovery or human cancer research or Mycobacterium tuberculosis genome-based antibiotics and identifying new ones and so on, there is so much you can do by converting your data or just looking at it from a different way, and that is in the form of a network. And so with that introduction, I'm really grateful to all of you for telling me about yourself. It helps me very much in any in any talk or any course to get to know the people we're talking to. It's the worst thing about the online world that we've moved into is that you're literally speaking at a wall, at a computer screen, of course, but it's quite a lot like a wall also. And so therefore, I am grateful that now I have some idea about who y'all are. And what I'll do is, now that we've had the first session part, the introductions over, we'll go to the PowerPoint. And so I'm going to stop sharing now. I'm going to reshare the PowerPoint screen so that you get to have a full screen view of what you want to look at. There. Hello, has it gone? Great. Do we see a full screen now? I think this should be better. Okay, great. So we've basically had the ice break, and I've got an understanding about who y'all are. What I would like you to do now, what I would like to say now is what networks are and how the network analysis and background began altogether. So this is already done part of this. There are diverse data sets that you have in biology. Most of you, as I just mentioned, you would be looking at spatial data or tabular data or textual data. Some of you would already be doing high-end, high-end genomics. So you would have genomic data, proteomic data, or maybe microarrays data sets in case you've not generated any of these data sets, you would possibly be looking at other people's data sets, those generated by your seniors in the lab or by others among your collaborators, or even public data sets. Okay, and therefore, it helps to understand that transcriptomes and RNA-seq information, the kind of data that we have available on the web, how it can be used and converted to a network. All of these data sets completely. Now, we in the lab are working on a unicellular green alga, the one you see on the right side right now. And this unicellular green alga is called Chlamydomonas reinhardtii. We call it clammy in the lab. Clammy alone, even though it's a unicellular organism, it's got at least 16 kinds of high throughput data sets. And to compare those data sets and contrast them and to overlay or superimpose them on each other is a huge thing if you start just going through the tables, because you've got tables with at least 17,000 genes in the nuclear genome, plus the genes in the organellar genomes, and the plastidial mitochondrial genomes, plus the transcription factors, plus the zillions of those that become proteins, plus the individual organellar, suborganellar proteomes, for example, the flagellar proteome and so on, and countless transcriptomes. So how do you look at all of this data together? And this is just a unicellular organism. Today, you've told me that you're all interested in human cancer research. So the kind of data you're looking at is far, far, far more, I would say, far more multi-layered. And therefore, it's extremely important to know how to reduce data dimensionality. Okay. Biological network analysis originated, it's not something new. Today we are taking this workshop and many of you have never used Cytoscape before, and we are in the 2020, but it is not new. It began at least 300 years ago. So it originated using the tools and concepts from network and graph theory and social network analysis, essentially. Originally, it was the social scientists who were doing network analysis, and then they moved on to trying and understanding, and then biologists took it over. Graph theory itself, for those of you who have read it previously or would like to use it in the future or understand it better, it's essentially a set of abstract concepts and methods that can be used to visualize and analyze biological networks. The first formulation of graph theory was done, it was done in 1700s, very early 1700s, when there was the problem of the 7 bridges of Kaliningrad. So there was this huge city, for which you see the map over here, and Leonhard Euler resolved it. So for going from one place, what is the route that you can find where you will cross? The city has 7 bridges, and how would you cross all the 7 bridges only once and yet be able to have a single route through it? So Euler resolved this problem, or this puzzle by understanding the idea of topology. He looked at the whole map of the city and then worked out individually what is the shortest path and how you would connect each of the 7 bridges. And that first thing that he did with the map in 1735 is the idea which set the basis for graph theory, the idea of topology, which all of us are now using. So in 1736, 35 and 36, of course, you had Euler working out the 7 bridges of Kaliningrad. And then, of course, for 200 years, people wouldn't care about it. In 1930s, again, sociometry began with Moreno, who first looked at flowers of hawthorn and tried to understand. This began with plant biology again, so it was kind of rediscovered. And then in 1940s, psychologists started using it, and they understood how cliques or communities might be formed. In 1950s, anthropologists came into the world of social networks. And then in 1970s, it was the rise of sociologists. And in 1980s came computational biologists, and that too, not true biologists, but computer scientists, essentially. In 1990s, adaptive radiation happened, and networks were used for many, many, many fields. In 2000s, then physicists and biologists descended, and now for the last 20 years, I think there's just no way, if you're a researcher, you can think about your whatever work you're doing, not just in science, also in social science, healthcare, medicine, finance. You've got to think in terms of a network. And so the field has literally exploded currently. And therefore, it makes sense for you to know what networks are, how to visualize the network, and what it means for you. With this, we start the second part of today's, tutorial. I will be introducing you to networks, topologies, and visualization software in this subsession. So what is a network? In the absolute basic fundamental schema, a network is just a grid or a series of interactions which have some nodes and some edges. So there are some nodes which could be circles or squares or diamonds, and then there would be some lines which are connecting those nodes. The lines that are connecting those nodes are called edges. So what are these nodes? When I say circles, squares, or diamonds, what are the nodes? The nodes are a set of actors. These actors could be individuals like you and me, people if we have to build a network of just our workshop today, what I would do is I would put myself at the center as the main node because I'm talking to each one of you. And then I would make, let's say we are 20 of us. So I will make 20 edges around me, and I will put each one of you at the end of those 20 spokes. So I'll become like the hub in a spoke model of a wheel. And so I'm the central node and y'all are peripheral nodes because you're listening to me. But some of you may be talking to each other. Some of you may be friends to each other. And so therefore there would be these edges connecting you with your friends or with others in the webinar or in this tutorial. And that is how, if you can visualize it in your head, we would form a network. If we are in a classroom, like I teach over here at NIPGR, and I also teach at Cambridge, there each one of the students knows each other much better than the teacher, than the instructor. So then y'all would be a much clicked or much interactive or much more interconnected series of nodes than just the teacher. The teacher would know you only to the extent of teaching you what I'm teaching you today, but you would know each other much, much better, and you would have smaller groups among yourself of friends who are closer friends to each other and others who are just acquaintances and so on. So a set of actors is the set of nodes. These could be individuals like people, but in molecular in molecular networks or biological networks, these nodes could be genes. These nodes could be proteins that are physically interacting with each other, or genes that are regulating each other, and so on. Similarly, a node does not just have to be one gene or one protein or one person. The node also could be a set of collections. Like if those of you who are working with plants, you might want to study multiple plant species. So your nodes could be species collections, or they could be countries, or they could be departments, or they could be genomes, and so on. And then you would try and understand the relationships between the countries, relationships between the species, between genomes, and so on. So you might be looking at a higher order level of network analysis or a limited order network. We will talk about different kinds of networks today, but the first idea that you need to get is that it's just a set of actors or collections, and they are connected to each other by something. So what are they connected to each other with? We call them edges, we call them lines. You could have wavy lines, you could have straight lines, you could have directed lines, you could have undirected lines connecting these nodes. So for instance, now in the classroom, if I'm the one teaching all of you, then the kind of line that connects me to you would be a directed line, like it would be an arrow going from me to you. So the arrow is focusing on you. But if there is a line connecting you and your friend sitting next to you and attending this lecture, then it would be a bidirectional arrow. So you are his friend and he is your friend. But in our case, between you and me, I'm your instructor, you're not my instructor. And so this is a unidirectional relationship. And this is extremely important to grasp as a concept, that relationships between nodes in a network could be unidirectional, bidirectional, or no directional, essentially. Then we'll try to understand that also. So a set of ties, so what are edges? Again, edges are a set of ties or relationships that connect pairs of actors. These could be positive edges and these could be negative relationships, okay? And each kind of relation defines a different kind of network. So for example, if we're just building a network of friends within the classroom, then it would be an acquaintance network. All of us are connected because we're acquainted with each other. If we build networks between you and your social groups, then we would have a friendship network. If we build networks or connect people who have written a paper together or co-authored papers together, then we would call them co-authorship networks. And that's what we do in in bibliometry. We look at co-citation and co-authorship networks in order to understand how a field has been evolving, at least in the published literature. And then, of course, if you're looking at the molecular data, and if you're a biologist, you would be interested in how two proteins are interacting with each other, and that would be a protein-protein interaction network. Or you may be interested in only knowing the regulatory landscape of your entire genome. So let's say you're working on Mycobacterium tuberculosis, and you want to know all the transcription factors in Mycobacterium that are targeting specific genes and how these are clustered with each other. So your interest is in the gene transcription factor relationship, and that would be a regulatory network. Those of you who are more interested in the transcriptome and they just want to know what part of the genome is expressing under a certain X condition, could be stress, could be developmental stage, could be the anatomy. Those of you who are looking at gene expression would be looking at co-expression networks and so on, okay? Now, why should you care about network visualization? I've talked about so many kinds of data sets that you might have in biology. I've talked about the kind of data you might be using. I've even talked about the kind of data other people outside of biology might be using in social media and social sciences. So why should you care about converting your data into a network? It's not just the elements of a system that matter, but how those elements are put together, you see? So more often than not, when you're looking at a table, and quite a lot of time, my first year students, they bring their entire data or share it with me as a large table. If you've got this huge table that you're looking at, I get lost, and you would more surely get lost because you're just starting with it. And one can completely get lost in the elements of a system and not be able to understand the elements of a system. What matters more importantly is how those elements are put together and how they are acting with respect to each other, and that you can never see if you're just staring at a tabular data set. So therefore, it's not just the elements, the rows and the columns of your network. What you have is essentially the relationship of the network. I'm sorry, where am I going? It's going too fast. Okay, with each other. Secondly, networks allow you to integrate diverse data sets meaningfully. They allow you to quantify relationships between variables and individuals, especially your samples, your experiments, your rows and columns. If you're looking at specific species or groups of species, you would be able to gain insights at species level, populations level, as well as systems levels. And then indeed, network analysis helps you find new patterns and create new hypotheses. We will understand every part of network analysis today, but more importantly, you need to understand that once you find these new patterns in any kind of data that you're looking at, it allows you to reformulate those patterns. And so therefore, it makes even more sense to build networks out of your data. So networks in biology, what kind of networks are we aware of in biology? I'm going to ask you to write in the chat box about the kind of network you might be thinking about. If you're an ecologist, and I know that none of you are ecologists today, but let's say if you were an ecologist, then you would be interested in food webs. You would be interested in how species interact with each other. You would be interested in fruit frugivory networks, how pollination is happening. So pollinator networks, ant pollinator networks, and so on. What are you, based on what your background is, what kind of network do you think you would be interested in? Please mention it in the chat box so that I am able to understand that you're getting the concept of building a network and a particular kind of network. So think about the nodes and the edges that you expect to be in your network. And if you're a neurologist, you would be looking at neurological networks. If you are looking at the metabolome, let's say you're a chemical ecologist, let's say you're looking at phytochemistry, then you would be, or let's say you're just interested in drugs. There was somebody who had mentioned that they are looking at drug vaccines. So you would be very interested in the kind of metabolic networks people have, or pathogens have, or their hosts have, and so on. Similarly, those of you who are just trying to understand the basic physiology of a system, your interest would be in signaling networks, for you to understand how these MAP kinases or calcium cascades, how they are connected with each other and how they are cascading and how reactions are happening one after the other inside the cell, particularly of interest to you in your species, in your physiological process. Those of you who are only looking at genetic interactions, you would be interested, for those of you who are working with specific sets of genes or gene families, you would be looking at genetic interaction networks. Or identifying novel or new transcription factors which are potentially regulating your pathway of interest in your species of interest, in your process of interest. So how do you find those transcription factors? You would be able to find those transcription factors if you are able to extract out a gene regulatory network from your data. Similarly, if you're only interested in proteins or maybe drug targets, then you would be interested in protein-protein interaction or protein-ligand interaction and so on. I do hope that some of you have responded, and I can see that many of you have responded to the kind of, oh, very interesting. Yes, indeed, the microbial interactions in the microbiome, these are enormous. 10 years ago, or even I might say 15 years ago, there was this huge interest in metagenomes, because the genomics explosion happened in 2000, and the human genome happily was deciphered just then. And so 5 years down the line, around 2005, everybody wanted to know not just individual genomes, they were interested in natural genomes. So they would just go and fill up a handful of let's say water from a lake, a cup full of water from a lake, or a handful of sand from your village. Just pick it up, and then you want to know what lives in there, what are the species in there. So we would completely sequence the genome of that handful of sand, or that cup full of water, or that cup full of ocean. And then we would say, all right, in a single drop of water, there are these millions and millions of microorganisms. How many different species are there? And we would sequence them, and we call them, because you can't call it a single genome because it's got potentially hundreds and millions, you would say it's a metagenome. But 10 years ago, and then in 2010, 2015, a new revolution came up in genomics, and that was the microbiome, when you discovered that you're not just a human yourself. So what you are is a walking, talking civilization of countless species inside you, on you, around you. From the moment humans are born, you're completely invaded by alien species around you. And so literally from your retina covering entirely every piece of skin that you have, it's not you, it's not human, it's somebody else inside you, in your gut, in your cheek. It's somebody else there too. And that's what makes you uniquely, because the kind of civilizations that live in the walking, talking you are different from the kind of civilizations that live in the walking, talking friend of yours next to you. And so therefore, this field altogether became the field of trying to understand the microbiome. So what is my, buccal microbiome? What's my gut microbiome? What kind of organisms live with me, on me, which make me me? And so to be able to understand that is a completely new field, and it's a very interestingly emerging field. I would say it's been running around for more than 5 years now, but it's got so much yet that is left to decipher. So those of you who are interested in the microbiome, fantastic. Those of you who aren't should be thinking about looking at it in future, and so on. So in cell biology, when you look at cell biology, think about a cell. Most of you are biologists, of course, and you would have done biochemistry in your kindergarten, right? And so the networks of cell biology that you have would have different players in these networks. You would have proteins, the proteins over here. So this is a very simple kind of network. On your left, you see the network as a drawing, as a real network. Here you have some actors. I told you there would be nodes, and there would be lines connecting those actors. So you can see different kinds of lines here and different kinds of actors and nodes here. The nodes could be circles, the node could be squares, and the lines could be orange or green or dashed or full or dotted and so on, or directional, directed, undirected, and so on. Each one of them will be a different kind of information. So you see over here, all of the square nodes that you have, like M2 and M1, they are metabolites. Can anyone name a protein in this network? All the proteins in this network are visible as circles. Can anyone name a protein in this network? And can anyone tell me how many proteins are there in this basic example cell biology network? Okay, you should count and let me know. And then, of course, there's also metabolism. So the kind of edges, you see the orange edges over here are the edges that represent metabolic interactions. The green edges, which are dashed, are telling you that there is a regulatory interaction happening there. The blue edges are protein-protein interactions that are going on. And the pink edges or the magenta purple edges are the cell signaling edges. And so regulation is a one-directed, is a directed interaction. So A regulates B or B regulates C. Can someone give me an example of X regulating Y in this network? Then I will understand that you have been able to read the network and decipher the network. So when I ask you, who is X that is regulating Y, you basically have to look at the nodes that have an arrow, a green colored arrow going towards another node, which is the target node of regulation. So the source node is the regulator and the target node is the regulated target, okay? Similarly, can somebody tell me any interaction in this network, which is a cell signaling kind of network? In a cell signaling kind of network, you would have a node which has a source, which is signaling, which is having a signaling partner or a downstream signaling node in another node, which is the target node of that one. So I would like to know if you can decipher the signaling partners here. And when I ask you, can someone tell me the pair of nodes which are doing multiple jobs? So definitely there's no such thing as one gene, one product, or one gene, one protein, right? Or one protein, one function. All of nature, all of biology is about multitasking. So can somebody tell me a pair of proteins that are multitasking? They're also involved in regulating each other. They're also involved in protein-protein interaction. Maybe they are part of a complex, and they are also involved in signaling. So let's see if you've got the right answer. Yes, absolutely. Absolutely correct, Shang, Sona, Zaved. Everybody's got the correct answer. Indeed, my idea is not to really test you. My idea is to let you know that it's easy, it's not difficult. And in this small network, if you can immediately decipher just by looking at the colors and the shapes and the designs, who is a protein, who's a metabolite, who's performing, who's interacting with each other, who's regulating each other, who's signaling each other, and so on, then you've already started to decipher a network. That means in your mind is already completely primed to do network analysis. So let's then move on to actually doing some network analysis. Of course, yeah, before that, you have to understand what kinds of networks might be there in cell biology. I had asked some of you to write down the kind of networks that you would be interested in. Here are some of these, such as undetermined functional associations, which you don't know of but would like to know. And then, of course, there could be co-expression networks that you're interested in. We are very interested in co-expression networks in our laboratory because we've got these time series expression data sets, and you can understand time series much better by understanding what sets of genes express together and then don't, and then are some of them regulators and are some of them, therefore it allows us to extract transcriptional regulatory inference from gene expression data. As a computational biologist, you have to learn more and more to adapt yourself to a knowledge-based approach. So it's not that you're going to be doing an experiment to find the answer. You're going to be looking at the existing body of knowledge to find patterns that others did not find before. And therefore, it's extremely important for you to know how to look at data and how to find patterns in that data. Then, of course, there are sequence similarity networks. Many of us don't think about sequence similarity. You know, if I ask you a question about how many of you have done BLAST in your life, when we start the course in computational biology, even in college level, we first teach a student how to do BLAST. It's the basic sequence similarity algorithm. Is there anyone in the group who's never done a BLAST here in our group? In case you've never done a BLAST, let me know. But I expect that everybody has at least done a BLAST. But when you did a BLAST, did you ever think about building a network out of your BLAST results? You could build very interesting networks out of your BLAST results. You could have done a BLAST of one protein or one gene of your interest with, let's say, a complete genome, or let's say 5 other genomes, or let's say the entire proteome. And you would have got some table as an output. Did you ever think of, and that table would have been such a huge table that you didn't have the time to look at it. I mean, it's like looking at it's got exactly as much panache as looking at a dictionary and being very interested about, not even a dictionary, a telephone directory and being interested about it. I would never want to look at a telephone directory. All you do is you look at the top ranks. Okay, so my top blast hits are these. But let's say you didn't want the blast hits, the top blast hits. You do always want them, but think more, think differently. Think about what else you could do with the data that you have, blast data. Couldn't you build a similarity network there? Couldn't it give you additional information on the kind of relationships you have with your, with the work that you're doing? Similarly, co-annotation networks and disease association networks. Those of you who are interested in cancer biology should think about disease association networks or building disease association networks for different kinds of cancers, or the particular cancer that you're interested in, and what kind of association it has, either with groups of people, which is population at the population levels, or either with groups of genes, which would be at the level of at the molecular level, and so on. Similarly, drug ligand associations and many more would be there. So when you look at a graph, the kind of thing you want to look at is the graph centralities or the graph matrices. And among the graph matrices and centralities, which are usually considered most important, which you should, I mean, there are tens and dozens of matrices and centralities, and there's just no way you can learn all of them. But if you're looking at a graph, you should be aware of certain centralities and matrices, which are important for you to understand the topology of your network or the kind of network that you have. The first of these always is degree. Degree essentially means the number of links that you have to a vertex. Now, if I'm the instructor in our classroom, and there are 20 of you, well, 25 of you that I'm teaching currently, then my degree would be 25. And you could separate this into in degree or out degree, essentially saying what is my in degree is the kind of number of people who are teaching me in this. So there's no in degree that way, but the out degree is 25 because I'm teaching 25 of you. Similarly, betweenness. So betweenness gives you an idea about the number of shortest paths that are there in the network. And we will see about it in the next slide to get a better idea about what shortest path is. But the shortest path in a network allows you to see how closely connected the network is and how easily, I would say, how vulnerable it is and how well you might be able to completely disturb it or perturb it. And that would be one way of looking at the shortest paths, calculating them, finding them for your particular gene of interest, and so on. Then you would have closeness. Closeness tells you the length that you have to all other vertices, or the distance you have as a node to other vertices. Then these can be centralities, closeness centrality, degree centrality, betweenness centrality. And then you would have densities of nodes, like the sum of the values divided by the number of possible types. So there are many different kinds of matrices and centralities. Suffice to say that you don't have to know them all, but it's important to know some important ones. And also many softwares, especially Cytoscape, will just find these for you automatically. So as long as you know that degree, betweenness, and closeness are important, Cytoscape will identify these for you. And then of course, mutuality these are higher order relationships, like motifs are also higher order relationships. What kind of dyads or triads are present in your network, and are they occurring more often than random? So that would give you some idea of how significant that value is for your network. The basic centralities in graph theory are, once again, don't forget, degree. So what is the degree of the orange node in the first picture that you see in front of you here? The degree of the orange node is, okay, I think my video is making my internet go down, so I'm going to switch off my video, but I hope that all of you can see the full screen and hear me clearly. In case there is any drop in connectivity, please mention it in the chat box immediately, because I don't want you to miss out on the most important parts. These are the absolute basic centralities that you must know about graphs. Degree. So I was asking a question, what is the degree of the orange node in the first network? Of course, it's 5. It's a simple question, but sometimes students get mixed up and they don't understand what it might be. So can you tell me, therefore, in this case, can you calculate in degree or out degree? The answer is no, because in degree and out degree, if you need to calculate it, you need to have directionality. From directionality, you would know that there is an outgoing direction to the degree and incoming direction to the degree, okay? Similarly, so very good, everybody gets full points on this one. Shortest paths, let's see. The shortest path from node A to node B is passing through 4 nodes, okay? I might say that you can reach from, this basically means that you can reach from node A to node B in a step of 4. Those of you who have heard of the 6-degree separation, you might have seen the movie about it also. The idea is that every individual on the planet is connected to each other by 6 degrees of separation, which essentially means that if we built a network of everyone, every human being on Earth, and we connected every 2 people who know each other, we would find that everyone is connected to everyone else at a maximum distance of 6, no longer than that. And most people are connected to each other at even lesser distances, which gives you one idea of how the whole world is essentially a family. And anyway, in the modern era, we are all a single empire of the earth. But still, the idea of shortest paths essentially tells you how long it will take to go. For example, do you see this node here up here in the corner and all the way here up here? Between these 2 nodes, I'm not able to draw them or mark my slide, but if everybody can see, this is a question coming along now, again. The uppermost node on the shortest paths network and the lowermost node, okay, there are 2 lowermost, but I'll take this one, the right one. What is the shortest path between them? Can anyone tell me how many steps it will take for you to go from this node to this node? I could take this as the first step, this is the second step, this is the third one, then 4th, then 5th, then cross over to 6th, and then go to 7th, then 8th, then 9th. Is that the correct answer? Is 9 the number of steps you require as a shortest path to get from the uppermost node to the lowermost node? Can someone tell me what it is? Alvaro says 5 steps. Let's see, is that correct, Alvaro? From here, if you took 5 steps, 1, 2, 3, 4, 5, no, you still don't reach this one. So can someone tell me what is the shortest path from? Yasmin is right, Yasmin, and Bhagyashri is right as well. EB is correct. Alvaro, no, I think you answered the previous question. I'm sorry, I think I mistook that for the answer to this question. So Wong, Bhagyashri, and Yasmin are absolutely correct. The shortest distance from this node to this node is 6 steps. I could have taken you round about in 20,000 different stories, and I could have taken you all the way here in 20 steps if I wanted, and I could have gone back and forth over some steps to make it 100 steps also. But that does not change the fact that these 2 nodes are truly connected to each other with the shortest distance of 6, okay? So you have a step of 6. And then if I have to ask you, what is the shortest path of the entire network? What would be your answer? Would your answer be 1, 2, 3, 4, 5? Or would your answer be 1, 2, 3, 4, 5, 6? The shortest path of an entire network. See, when I give you 2 nodes, this node X and this node Y, between them, you just had to calculate the shortest distance and you told me what it was. But if I give you any other nodes, like node A and the blue node next to it, what is the shortest distance between the node A and the node next to it? You don't have to type the answer. It is 1. You can just get to it in the next step. So the shortest path is 1 for these 2. The shortest path is 5 for A and B. And the shortest path is 6 for what you just calculated between X and Y. So therefore, when I ask you the question of what is the shortest path of this network as a whole, what would you say? Can someone guess what is the shortest path of the network as a whole? The shortest path of the network as a whole is the shortest distance required to go from any node to any node. So that means that the moment you say this is the shortest path of the network, let's say 3, then in 3 steps, you should be able to reach every node from every node, at least in 3 steps, if not lesser, okay? So for this network, what is the shortest path? In how many steps will you reach from everywhere to everywhere? And that's why they say that humans are separated by 6 degrees of separation. I personally don't believe it's 6, it's even lesser, but let's say it's true. I wouldn't know somebody studying in Timbuktu, but if you're attending my class today, then it immediately brings us down to a separation of one. Either way, so the shortest path in this network is actually the longest shortest path, and that is the one you just calculated, which is the number 6. Absolutely correct, Yasmin. So when you say 6, because you've already calculated the longest path between the farthest nodes, no other nodes would be that far away from each other. So obviously you've already taken into account the farthest nodes and their shortest path. So every other node on the network would be either that or less than that. It couldn't be more than that because no other two nodes are farther away than the guys you found as the farthest nodes. Okay. Well, there might be. What about A and this guy at the end? 1, 2, 3, 4, 5. Yeah, that's 6 too. Perfectly right, Yasmin. Absolutely correct. So we will look at scale-free networks. So most biological networks, because you're all biologists, you will be looking at different kinds of networks, but most biological networks are scale-free networks. And when I say a scale-free network, it basically means somebody that follows the power law. When I say the power law, this is what the earth runs on, or what humanity runs on. Rich people getting richer, poor people getting poorer, and that's not something you can blame on anybody. It's just that it's almost like a way. It's basically saying that if in a network there's one person who's connected to many others, basically that person might be the teacher. In our network, in this classroom, I'm the instructor, so I'm connected to most of you. So if you just go by richness of degree, my degree is highest. If a new student comes into the class, do you think that student would connect to one of the 25 of you, or do you think that student would immediately connect to me? Obviously they would come to the class to listen to me. So what will happen? My degree will increase when somebody new comes in. That somebody new will not be thinking about, making the world a more equal place, that she's the richest, let's not make her more rich, let's just make the poorer people richer, and they would go and connect to you maybe and start learning from you. It doesn't happen like that. So therefore, the power law is what is the real world system. So real world networks follow the power law, and real world networks are therefore scale free. And so there are some nodes which are highly connected and other nodes which are not so connected. Just like that, in a cell or in a biological system, there are some nodes which are very important, and it might be constituted of genes, it might be constituted of proteins, or it might also be transcription factors that are regulating a large number of targets. So if a new gene comes in, it would still be regulated by one of the transcription factors. So essentially, the rich keeps getting richer as the network expands. It does not change things. So what's transitivity? So that's a scale-free nature of a network. When you look at a network, if you make a random network, you will find that the random network generally is less scale free. So it doesn't follow the power law. Randomly, you will find things falling into an equilibrium. And the moment an equilibrium comes, it basically means that everything is connected to everything in much the same way. And that is not a real network. That is not biology. The moment you find a non-scale free network, you know it's unreal. It's a random, or it is a model, or it is just designed by somebody wrongly. So then you have transitivity. Transitivity is the next centrality, which is also extremely important. It gives you an idea about how much a set of nodes lead the way to other parts of the network. So how much transition you can have through them. And the last one is centrality. How are some nodes central? Nodes that have greater degrees are obviously central, but nodes that have less degrees can also be central. For instance, over here you can see centrality has been given as a score to each node to the network, the last network that we see. The most connected node is the red node, so it has the highest centrality. The reason it has the highest centrality is that it is connected to all parts of the network. There are three. If you really look at the network from out of the box view, you will see that the network essentially has three parts: part 1, region 2, and region 3. And the red node is connected to all three regions, and therefore the centrality of the red node is very important. It could be that the orange node here was connected to many other nodes and had a greater degree. But even though it might have had a greater degree than the red node, the centrality of the orange node is lesser. Why is the centrality of the orange node lesser? Can someone tell me? Both of these, there are three regions to the graph: the upper right red region, the orange region, and the other orange region. However, the highest centrality has been given to the red node in this network, and the second highest centralities have gone both to the orange nodes in the network. Why were these two also not red? Why did they not have the same centrality or as much importance in centrality of the network as the red node? Can someone answer that question? I hope you can see the network clearly. Only then you would be able to answer the question. Think about it. Why is red more central in this network rather than orange nodes? Orange nodes are central and they have a good centrality score, but they are not as central as the red node. The red node is most central. The reason here is that the orange nodes are not connected to all parts of the network. See, this orange node is only connected to its own region and the region of the red node, not to the third other orange node. Similarly, the other orange node is also not connected to the first orange node. It's only connected to the red node. The red node, whereas, is connected to itself, of course, as well as both of the others. And so therefore, the centrality of the red node is much, no, Yasmin, you gave the wrong answer. Red node does have a high degree in this network, but it is not the most central based on degree. And that's the most important concept I'm trying to make you understand over here. Wong is right. The reason red is the most central node is that it's connecting everything. Or it's also technically that's not true. Red is not connected to everything because of course it is not connected to green here or there, but it is connected indirectly to everything. Similarly, orange is also connected to everything indirectly because this is totally a connected network, right? And yet orange has less centrality. It's because red is more central in the sense it's more directly connected to everything. So orange is connected to everything. Every node is connected to everything. But which ones are more directly connected to everything? So a degree would be important. The degree would be a main feature for identifying centrality, but also the presence of that node in the network. So the presence of that node in the network has to be more central, more closer. So closeness centrality to other nodes is more important. Okay. So a quick primer on network analysis is where you get to know topologies. When I keep repeating the word topology, nobody has yet asked me, what is topology? Topology is the landscape, right? When you think about the topology of the ocean, what's the word that comes to your mind? Flat. When you think about the topology of Mount Everest, what's the word that comes to your mind? High. Undulating, Himalayan range, Sino-Indian border range. It's all the Alps, for example. So it's undulating topology. So therefore, the moment you look at a network, you get a word in your mind, and that word is the topology for that network. Now, for example, here on the left side, the leftmost topology is a ring topology. Why would I call it a ring topology? I call it a ring topology, not just me, the world calls it a ring topology because every node is connected to every other in a daisy chain, and the last node is connecting back to the first node. And so the first word that comes to your mind when you look at this topology is it's a ring topology. Each one of these nodes could be individuals, they could be friends, they could be, they could be genes, they could be proteins, they could be species, but whatever they might be, the topology is still a ring topology. Then the second one is a mesh topology. The third one is, of course, a star topology. Now, some networks are fully connected. When you say a fully connected network, that itself becomes a topology because very, very few networks are fully connected. Biological networks will usually never be fully connected. I told you they are scale-free, right? So a fully connected network will never follow the scale-free topology. And so therefore, these are fully connected networks. And more random networks, when you try to build models or when you design, computationally, you come up with new networks. Sometimes you want to work with fully connected networks. And these have their own features in very interesting patterns that you will find when you study fully connected networks more carefully. Then, of course, there's a line network. The line network and the ring network are very similar. Only difference is that the first node is not going back and connecting to the last node. So that's your line network. Then, of course, you have a tree topology. Tree topology is where you have a branching dendrogram kind of view. So the branching dendrogram essentially is telling you that the tree is basically has got a root node, and then that root node diversifies into two branches, and those branches further diversify into more branches. So this is a tree topology. And the last one is a bus topology. The bus topology is particularly interesting because it's basically everyone is sitting around the line and on different sides or different windows in a bus, like almost in a bus. Bus topologies are found more often than not in systems, in institutional, networks like LANs and WANs. So I've been the system administrator of the NIPGR network for, I had it for the first 10, 11 years. And we were thinking about what topology would be the best to fit when we were designing the network for the institute. And of course, we ended up framing a combination of star and bus topologies. And it turned out to be, it's been quite successful even now, while I've moved on to Cambridge and done other things. So therefore, there are different kinds of topologies that you should be aware of. And you should also know that topologies are never single standalone. So they could be networks which are combinations of these topologies. Okay, let's take a little break while I close my door. I think there's a lot of noise coming from outside. Just a second. All right. Yeah, this is quite, it's better. So these are the common topologies. And how about looking at robustness against random failure? So when you've got a network, when you've got a particular kind of network that you're looking at, why should you be looking at it? I did talk to you about it earlier, but let's say this is the network you're working with. Let's say you're an ecologist, or let's say you're a cancer biologist, or let's say you're a drug developer, and you've got this network to look at, okay? What does it help you with? What does it tell you? The first thing you would want to know is, how stable is my network? If this is a pathogen network, let's say this is a disease network, then the first question you should be asking is, how should I destroy this network, or how should I break this network apart? But if it's your host encoded RNAi network, then you would not want to destroy it. Then you would want to know, how do I protect this network, and how do I keep it safe against failure? And that failure could be targeted failure or random failure. So you would want to know the robustness of your network. So let's see how robust is this network. In this network, let's say you remove one of the nodes randomly remove any one of the nodes. Okay, let's remove this guy. So the peripheral node has gone. How do you think this network has affected? Would you say this network is still okay? It still looks okay, would you say? Or do you think we've destroyed the network? What's your answer? Does it still look okay to you, or do you think we've really damaged it badly? Yeah, it still looks okay. Perfect. Now again, look at the network at this stage. You've removed another 2 nodes of the network, and the network still holds on to its basic structure, which is a singly connected series of nodes, all of them having edges connecting to each other. So from any one node, you can still reach any other node. Even though you've removed 3 nodes from the network, it is still reasonably robust. It is still reasonably okay. Now imagine in the starting network, you just removed 1 node, which was the central node. Okay. The moment you removed the central node, what did you do? By removing a single node, the central one, the hub, you might say, or the highest degree node, and this is a very simple example, you basically broke the network into pieces. You fragmented the whole network. And when I say you fragmented the network, you've essentially destroyed it, because you've broken it. Exactly. So you've completely broken it into pieces. And by breaking into pieces, it means that you've stopped the interconnectivity between this part and this part. So earlier, if the message of destruction could have gone from one part to the other for the nodes to find backup, if they were genes, they could have got backup genes, they could have activated other genes which were lost, and they could still have reformed themselves. But now these nodes no longer can talk to each other because they've become disconnected. So and by killing one node itself, or attacking one node, you've also left 3 other pieces of nodes disconnected with everybody else. So they've died out too. You see? So if you're looking at an ecological network, and this is a very important feature of ecological extinction cascade. So when one species goes extinct, we don't really think about it as a single loss. Along with that species, many other species also go extinct. You see, in Australia, I don't know, obviously there's nobody today from Australia, and therefore I can give an example. For in Tasmania, for example, for tens of thousands of years, humans had lived there when nobody had come. and they were living there just fine. And then, of course, James Cook expedition happened, and the British discovered Australia, and then they took over Tasmania, and the kinds of species that were there in Australia, Tasmania, and the Oceania, as humans started to take over and claim these continents for themselves, species started getting destroyed. Dodo was one of the first. Nobody even knows how it looked like right now. We just have a few pictures, but nothing real. When these species went away, along with them, if it was a tree species, then obviously the animals that depended on it were also killed off. If it was an animal species, then every plant that depended on that animal for its pollination or for its seed dispersal was also killed away. And therefore, one node is not just the destruction of one species or the extinction of one species, is never the story of extinction of a single actor. Many others get secondarily extinct along with that. So secondary extinctions and primary extinctions is a major issue in ecology. And we are very concerned about secondary extinctions that happen by targeting one node that you've actually removed, and you end up removing many others which you had not planned to. And that is what happens even in genes, even in proteins. And so therefore, it's very important for you to know, therefore, the graph centralities that I spoke about earlier, and within those centralities, how attacking a node would affect the rest of the network. And so therefore, if you were somebody, and if this was a disease network, then you would think about planning to destroy which one. Then you would probably target just this network. So let's say now you're a researcher, and this is the network of how HIV, or let's say this is the network of how the coronavirus is affecting your immune system, and you've been able to find some important genes and proteins which are working in the COVID network. Which node would you attack if you had to destroy this network? And please don't answer the central hub, because that is not the answer. That's not the correct answer, because when you have a network like this, obviously the node with the highest degree or the greatest hub centrality or closeness centrality is also the one which is most protected by that pathogen. So it would have multiple backups, a huge set of gene families, a huge set of backups. If something happens to this, immediately it is replaced. So you can't really affect it, because the system knows that this is a vulnerable point, and it would have the maximum protection around it. So this is the most vulnerable, but also the most protected hub. So where would you design your attack if you had to attack the system, and you knew that you couldn't go for the highest straight for the prime minister, because he would be protected? Who would you first destroy, to know that you can still break the system apart and still damage it, and then maybe try and get to the prime minister or get to the most the greatest hub? You don't know about the backup, Shofi. What happens is, we don't know if this has 100 backups or 20 backups, and it's better not to attack something that you don't know enough about. All you know is this is hugely protected with backups. So we can't, if we knew anything about the backups, we would have them in the network. So is there a node that you can still attack? Yes. Who is this? Alvaro. Alvaro, you're absolutely correct. And Wong is also the correct. Similarly, Yasmin, what we would do is take this one. So there are at least 4 nodes connected to the main hub. And out of them, there is one which has greater transitivity. You see this guy, it is more transitive, and it is more between central, because it connects one region. It is the only bridge to the connection to the rest of the network. If you destroy this one, you're basically removing 2 others, but not cutting out connection. You're not breaking the network into 2 parts. But by removing this node, you would be able to fragment the network into 2 parts and disconnect the network and the communication that possibly the COVID virus requires for its sustainability. And even though there's a huge backup for this central node, the virus may not have a backup for this guy, because it is less visually important, but still it is quite critical, and you might want to think about how best to destroy this. Either this relationship or this relationship should be targeted, and you would have to come up with ways to remove that. Similarly, if you're protecting this, if this is your network of the host encoded host how would you say, host encoded self-immunity. In that case also, you would of course be very careful about this node because this is an important one, but you would try to protect this also, because you know that both of them are equally important, if not equally, but quite critically important. And this, the importance of this second node becomes even more because this is usually the target of external invasions. And so these are things that networks teach you only by practice. So there's stuff that you learn about randomness and robustness and vulnerability of a network and the extent to which it can sustain failures. So in what we'll do next is to see the types of networks. We'll look at small world networks and random networks, of course. I had taught you just now, I was talking to you about scale-free networks, right? And scale-free networks have this unique feature of being small world networks. So some nodes which are hugely connected to a large number of huge degrees, and others which don't have a lot of other connections. So in general, any two arbitrary nodes are connected by a small number of intermediate edges in small world networks, and the network has an average shortest path length, which is much, much, much smaller than the total number of nodes in the network. As against this, a random network would be generally homogeneous. You can see it here, and the nodes would have quite similar degrees, and they would not be robust to arbitrary node failure. So if you arbitrarily remove any node from a random network, you have a good chance of fragmenting that network and damaging it very strongly. But if you just take an arbitrary node in a real small world biological network, there is a chance that you cannot harm it because there are only few protected, safe people. Everybody else is dispensable in the network, or not really, I shouldn't say dispensable, but still the network is hugely robust to random failure. And that's because it's got a much shorter average path length. Secondly, the interaction networks that have, that are seen in proteins or in genes or in species or in biology, they have been shown to be small world networks. And the degree distribution of such a network always follows the power law. And that is what I was telling you in the beginning when I was teaching you the power law, that it's the moment you look at the degree distribution, and you should, whatever network you have, the first thing you should ever do with it is to build a degree distribution of it. And then you see that it is really following the power law or not. Is it a small world network or is it a random network? So this is the distinct difference between the, so if you look at a random network, you would have a Poisson degree distribution straight out. And so therefore, there are many algorithms that exist to create random networks for you. You can just tell your random network, okay, I want a random network of 100 nodes, and it should have, let's say, 1,000 edges, or 100 nodes and maybe 50 edges. And there are different kinds of algorithms that are going to build them for you. You could build random scale-free networks, flat networks, small world, high clustered networks, and so on. Each one of them is called different things, but these are more intermediate. In the next course, which we have on intermediate networks, you will understand each one of these kinds of networks. Today, I want you to remember the basic concepts. Just based on edge properties, what would you call your network? You would call it either undirected, directed, or weighted. Now, by looking at a network, you should be able to immediately recognize, just the way I was saying, by looking at a network, you should be able to recognize its topology. Is it a ring? Is it a mesh? Is it a line? Is it a bus? Is it a star? And so on. Is it fully connected? And so on. So based on edge properties, you should be able to recognize a network as undirected. The blue network here is undirected because there are no directions between the nodes. The green network is directed because you have nodes that are pointed at other nodes, and the directions could mean just about anything under the sun. But let's now not worry about what they mean, because they would mean something based on what you're looking at, what your interest is in. Main thing is they are directed. And the third network is the weighted network. Both directed and undirected networks may be weighted or unweighted. In this case, the left two networks, the blue and green, are both undirected as well as directed, but they are unweighted. And the third network, which is purple, is undirected but weighted. So when I say weighted, it means that each edge which is connecting the nodes has got a weight to it. So it's got a value there that is given to it. So that value could be connected based on, that value could be anything. In case these are genes and these are expression values, so this could be co-expression and so on, okay? The other basic feature you have to know is that every network is essentially a matrix, as a graph, because this is graph theory. So all networks are generated from graphs. The first blue network is generated from this graph that you see below you. And these graphs are called matrices, and these make the real name of the matrix is adjacency matrix. So your adjacency matrix is what you will need to have if you're using R. You will need to build an adjacency matrix from where you will get the actual network layout. So these are layouts, the blue layout, the directed layout, the weighted or unweighted layout. But the true network, the actual data is sitting in a graph, and that graph or table is where you begin with usually. So your data is currently in the form of a table. From the table, you will have to generate a matrix. From the matrix, you will have to generate an adjacency matrix. And from the adjacency matrix, you build your layout. And that's how it is here. Please take a look at this and think of questions if you have. I will take a short 1-minute break just now. Alrighty, I'm back. So has everybody been looking at the adjacency matrices and the networks, and do you have any questions in mind? Very good. So how do we distinguish directed or undirected from the adjacency matrix? Very good question. I'm just going to tell you that. And second, how to count in and out degrees in the network or in the matrix. Very good. So what we'll do is we'll try to understand the first one. The first is an undirected network, and this is an adjacency matrix that builds the undirected network. So here what you find is every cell has a value of 1 or 0. So this is a binary network. The moment you say it's a binary network, it is therefore an undirected network because you have only one of two options, yes or no, which means that either two nodes are connected, yes, or two nodes are not connected or disconnected. That means no. So when you have only two options and when you have only 0 and 1 in your values, then straight away from the adjacency matrix, you can make out that this is an undirected network because all you know is whether or not the edge is there. You know nothing else about that edge. Similarly, in the next one, again, it's a 0 and 1. So you see there's nothing except 0 and 1 in the cells, which means that all the cells are yes or no. Present, all the connections are yes or no, which means the connections are there or not there, which means that it is an unweighted data. In the third data set, look, do we have 0 and 1, zeros and ones here? We do have zeros. We don't have ones. So instead of a yes, you're not telling yes. You're saying yes resoundingly, or yes lightly, or yes maybe, or yes surely. So there are different ways of saying yes. And the moment you bring in different ways of saying yes, you've brought in a weighted network. So what you have here is you've got your edges which have scores of 8 or 12 or 16 and so on. And the moment you bring in these values which are not 0 or 1, which are not binary, you've basically brought in weights. So that is how you read out immediately a binary or a weighted network and an unweighted or a weighted network. So the first two networks are unweighted because they have a binary data information, only yes or no. And the third one is a weighted network because it does not have binary information, but weighted information. So now how do you read this information? Let's go back to the first network, which is the adjacency matrix. It is a binary data, 0 or 1. But what does 0 mean and what does 1 mean? I've told you 1 means yes and 0 means 0 means no. But still, so A and A have a value of 0, which means that A is not connected to itself. A and B have a value of 1, which means that A is connected to B. And so we begin with the node A and we don't connect it to itself. That means we don't make a loop around it. Then we see that there's B and there is 1 value here, which means that A is connected to B. Similarly, A is connected to C because there is a value of 1 here. Similarly, A is connected to D. And so we keep increasing these. A connection to B comes in, A's connection to C comes in, A's connection to D comes in, and so on. Finally, we find that A is not connected to G. Look here, A to G is 0. And so you see in the network, A is not connected to G. Is G connected to anybody? Does G have any value of 1 in its own network? If you look at G, G is 0 with everyone. That means G is not connected to anybody. Hold on, right here, G is connected to somebody. There's a value of 1 that G has in a cell that matches the column F, which means that G and F are 1, which means that the relationship between G and F is a yes, which means that G and F are connected to each other. So this is how you read the adjacency matrix and find the network or draw the network. We call it adjacency because it tells you whether or not two nodes are adjacent to each other. Adjacent means connected. So it's a connectivity matrix. It basically tells you yes or no for every single pair of genes. Can someone tell me the value of B and G and why B and G are not connected in this network? You see, B is connected to F and B is connected to A, but B is not connected to G because B has only 2 yeses or 2 ones. The first is to A and the second is to F. So therefore, B is connected to A and F and not to the others. In this case, in the second network, which is a directed network, can somebody tell me if B is connected to A and F? Again, over here, B is connected to A and F. Is B connected to A and F here also? You see, when you go into B, you find that it is connected to C, it is connected to D, it is connected to F, it is also connected to G, but it's not connected to A, which means that B, hello, it is. Also, that's because it's directed. Can someone tell me how you can distinguish directed from an undirected network? The way you can distinguish the directed from an undirected network is that the diagonals are not the same. In an undirected network, the diagonals are identical. So when I say who is connected to B, you will just read either the row or column. You can just go to the row B and you can read it out. B is connected to A and B is connected to F. Similarly, you could just, instead of reading the row, you could have just read the column. B is connected to A yet again, B is connected to F yet again. So whether you read the column or the row, you get the same, absolute same information. And this is what means you're looking at an undirected network. It doesn't matter whether you're looking at the row or column, but here it matters whether you're looking at the row or column. So when you're looking at the row, B is connected to C, D, F, and C, but not to A. But if you look at the columns, B is connected only to A and not to the others, C, D, F, C, which is why the lower part of the diagonal is completely different from the upper half of the diagonal, and which is what tells you that this is not a simple undirected network. It is a binary network, it is an unweighted network, but it's got a direction. And that direction, the arrow, the sense comes from the upper part of the diagonal or the lower part of the diagonal. Okay, so there's a question saying the non-diagonal for directed network would never be symmetrical. Yes, the two sides of the diagonals would not be symmetrical. So when you have a directed network, you would never have the upper part common to the, in any kind of square matrix or adjacency matrix, usually the diagonals are identical. But in case of a directed network, it is not necessary. It could be that B might be bidirectional. And if it is bidirectional, then on the row, you will find a 1 for B and A, and you would also find the column with B and A together. In those cases, special cases where there is bidirectionality, you might find that the upper part of the diagonal gives you the same information as the lower part of the diagonal, but it may never happen always, because direction, the moment you say it's a directed network, you cannot say whether it's a bidirected network. There might be some pairs of genes which are mutually regulating each other. It's highly unlikely, but it might happen. So in those cases, you might have similarity, but never take it for granted. So as soon as you see that two parts of a diagonal are different, even in one single cell, then it is a directed network. Now, how do you distinguish a directed, undirected, and weighted network? That I've told you already. The moment you see the weights, you know it's a weight. So the next question was, how do you read the out degree and in degree? Here you have, when you have the degree of A on the rows, you just calculate the degree of A. Over here, you calculated the degree of A as 1, plus 2, plus 1, plus 1, plus 1, plus another 1 and 1. So it became the degree 5. And that was true. A was connected to 5 other people. In the directed network, if you just look at the row, the degree of A is 1 because it's only connected to B and nobody else. So the out degree is read on the rows and the in degree is read on the columns. So when you go to the in degree of A, can someone tell me the in degree of A? A is not connected to anything. And therefore the in degree, that means incoming to A, the number of nodes that are incoming to A are 0. The number of nodes that are outgoing from A are 1, and therefore the out degree of A is 1 and the in degree of A is 0. Can someone tell me what is the, by looking at this network, you're looking at F in front of you, here's F. Can you tell me the out degree of F and the in degree of F? Out degree and in degree of F is what I'm asking. Let's see. So when I say out degree, it basically means how many arrows. Yes, absolutely correct, Yasmin. So in degree is 1 and out degree 0. Is there any node in the entire network which has a non-zero in degree as well as a non-zero out degree? It should have both. It should have a non-zero in degree and it should also have a non-zero out degree. Can you name such a node in the network? So like A has a 0 in degree, F has a 0 out degree, G has a 0 out degree, but I want a node which has non-zero status on the in degree and the out degree. The node B. No, it would not be an isolated node, Yasmin. A non-zero. I'm saying non-zero. If you have a zero in degree and out degree, then you have an isolated node. Absolutely correct. But my question is, listen to my question again. I'm asking which is the node in the network which has a non-zero in degree and a non-zero out degree. So anything 1 or above in the in degree and anything 1 or above in the out degree. So the answer that Abhirupa has given, Abhichandan has said D. You're absolutely right. D has an in degree and an out degree also. But the true answer is what C. Wong and Abhirupa has given, that there are 3 such nodes. You look at B. B has in degree and out degree. C also has. D also has. So B, C, and D are the real answers to the nodes that have all 3 kinds of degrees. Similarly, this is a weighted network and you can read off the weights. And this is a very simple weighted network because still you see so many zeros. But in a true weighted network, like we have the co-expression network data sets, you have zillions of, like thousands, tens and thousands of cells in the adjacency network, which are full of values, and they are decimal values to n number of places, so and so on. Okay, biological networks. So we've already read types of, I think this is a repeat slide, sorry. So there are different kinds of biological networks which you are aware of. There are different kinds of networks that we are working with in the lab. We look at phytochemical networks because we are working with ecologists. We also look at photosynthetic networks. This is Situ's own work that she has presented at InCoB. Situ already has presented an oral presentation. I think she did it yesterday, where she presented photosynthetic networks, temporal light and dark networks that we built for different stages of Chlamydomonas reinhardtii using undirected and directed methods both. And then, of course, we look at network. We've built a network perturbation algorithm in the lab, which is called Nextcade. It's all free and you're welcome to use it. Whatever work we do in the lab, we try to make it open source so others can use it. And so my next session is on creating and visualizing your own networks. I wonder if we should, Shafi, should we take a break? I want to be able to give another 15 minutes to talk about how you create and visualize your own network before people do it actually. So I want to show you how it is done, and then I will make you do it, because I don't want you to start from scratch. It usually becomes very complicated when you're doing something for the first time and somebody says, okay, do it now. So I'm going to present it to you, but we have 15 minutes before our break. So I'm going to, essentially, oh, fantastic. I got a great news, made my day. So coming back, okay, back to it. So sometimes you have your phone in front of you and there's a message that you see you're not meant to be seeing, but I just saw a message that we've got sponsorships for all our seats on the our workshop that we're doing in December. Oh my God, I can make the seats free for everybody. This has never happened to me before. Great. So it's thanks to InCoB. I think it's my good deed today that has turned out to be such a good news. Oh, thank you. I've been waiting to get sponsorships because you don't want to do courses where students have to pay, and if you can make it free, it's so good. Great. So let's come back to Earth. I'm just feeling I was worried about it. So how do we calculate the degree for weighted networks? That's a very good question, Wong, and I think I missed it. So I'm going to go back to the previous slide and just look at it for you. How do you calculate a degree for weighted networks? You calculate a degree for weighted networks by adding the weights of each edge that goes out of that network. Okay, so you see for A, over here, A is actually connected to only 5 other nodes in this weighted network, but it is connected to those 5 nodes by 5 lines or edges, and those edges are not the same. And therefore, you have to add the sum, you have to sum the weights of those edges to find the true degree of A. The true degree of A is not 5. It is, because it's a weighted network, you will have to sum the weights. So the sum of weights is 8 for 0 for the first, because A is not connected to itself, plus 0, plus 8, plus 12, plus 12, and note this, A is connected to G with 12. G is connected to A with 12. So there is a connectivity between A and G also. So you will connect, because it's disconnected. I think over here, what is happening is that the weight between F and G, let's look at the weight between F and G. The weight between F and G is 12 over here. And the weight between A and G is shown as 12. But I don't think this is it. I think you can't do that, because A and G should be disconnected. They are not disconnected in this. So this is a small error. I apologize for this. This should be 0. And then you have to actually calculate just the addition of these. So let's do it for B. For the node B, your weights are, B is connected to A and B is also connected to F. Okay. So its weight to A is 8, which is very thin, but its weight with F is even thinner. It's a tiny line which you almost might miss if you don't look at it carefully. So the total degree of the node B in this weighted network is 8 plus 4, which is 12. Okay, so I should fix this for my next tutorial. There is no connection between A and G, or I could just add a new edge between A and G, and then we would have all of this matrix coming out to be right. So very good question. And moving on to the next part of the network, because I want to be able to be done with it. So now that you know a network, now you know how it looks, and you also know how to extract it from a given adjacency network, what kind of tools would you use to visualize your network? My always first priority is Cytoscape and R. In R, you have many packages like iGraph, SNA, Social Network, and many others. In Cytoscape, you have over 200 different plugins, but mostly 60 or 70 are used, which you should be aware of. But don't think that just because I'm talking about R and Cytoscape, these are the only tools in the world that you will use to do network analysis. A large number of statisticians use things like Gephi. and Pahek, and a large number of mathematicians. They would swear by Pahek and they would not take anything else in the world. But then again, some of my own colleagues have built other network visualization tools like biological networks, Bayana, and so on. So you would be, sometimes some of these tools are used by people in specific areas. For example, if you're looking at a hierarchical complex system, and if you've got zillions of nodes, like you're looking at the entire Twitter network of the world, you would need, first of all, you would need a heavy workstation machine, a server possibly, to handle that kind of a network. But then you would also need a network visualization tool that can really grasp all of it together. So Gephi is definitely one of them. Cell Illustrator, Medusa, Navigator, NIAT, Pathway, Pivot, ProK, ProViz, PabView, Pina, and this list is not comprehensive. This is just, maybe you're looking at the tip of the iceberg, but there are more network visualization tools in the world, which should just give you only one message, that if you've not started looking at your data as a network, do it yesterday. I mean, do it right now. So you've got to be there. It's already available for you. Most of these tools are free. Don't lose out on what is available to you to use. We are going to begin with the open source cross-platform huge consortium called Cytoscape. It has, the best thing about Cytoscape is that it takes inputs which could be either a network itself, or you could give it a table. And sessions will save everything as CSS files, networks, tables, styles, screen sizes, even hundreds of plugins for analysis of specific kinds of data sets. And you could export your network in different formats like CIF, GML, XGML, biopacks, whatever else you want. And you could export the graphics in very high publication quality figures, as we will show you today in the stepwise tutorial. Given the time, I'm going so slow, but please let me know if you really think I'm going very slow. I will quicken my pace a bit, but I realize that some of you have never used Cytoscape before, and or done network analysis before, so I've been a little bit slow. If you feel that this is too slow, I will be more careful, or I will try to rush through things. So Cytoscape has many special features. It's got network topology statistics, motif analysis, and modularity analysis, network alignment and comparisons you can do in Cytoscape. And let's begin with a very simple kind of input data. So let's begin with people who know each other. Now, I'm Gitanjali Yadav, so I'll call myself GY, and I'll become the one node here in this network. And I'm building a network file. So you should do this on a text. Whenever you're building it, don't do it now. Please don't do it now. We'll do it together. But right now, just pay attention that you need to build a text file in which you will have your nodes GY. And let's say I want to build a node about my social network. So the first group of people I am connected to are my family. So there are 3 members in my family, AA, AB, and AC, whoever they might be. And I also have 3 very close friends. So those friends are X1, X2, and X3. And I also have 3 students who are working with me in the lab. So I have a group, a professional group composed of 3 individuals called FO, FM, and FP. Oh, somebody needs to pick up the phone in my office so that I don't get disturbed. Thanks. So we have 3 sets of nodes here. The set of A nodes are my family. The set of X nodes, X1, X2, X3, are 3 people who are my immediate, close friends. And the set of 3 people, F, O, M, and P, are people who are my colleagues at work. These 3 sets of people, or these 9 people, I know them all. So what are the edges that the network will form? I will form a connection GY with AA. That's the first edge, because I know AA. I also know AB. I also know AC. Okay. Similarly, GY to X1, X2, and S3. Similarly, GY to F, O, FM, and FP, madam. So yeah, so these are, I've made it K, O, K, M, and KP, but essentially 9 edges. Do you see? So these 9 edges become my text input file format. We call it a simple information format, a paired format, where you say GY is the first node, GY is connected to AA as the second node, and the relationship between them is 1. I could give it any number or any character, but the relationship gets a value. And the moment you give 1, I prefer to give 1 because I showed you on the adjacency matrix, 0 or 1 is a good way of showing a binary relationship. So my relationship with each one of them is a yes, and therefore it's a 1, 1, 1. Another way of writing this could be What's the difference? Yeah, another way of writing this could be that my relationships with each one is different. So with my family, my relationship is something called 1. With my friends, my relationship is something called 2. And with my colleagues or students at work, my relationship is something called 3. So all of you would also immediately therefore understand that there are 3 sets of relationships or edges in this network of GY, let us say, and there are 3 kinds. But there is a major mistake that I have made over here. Can somebody grab the mistake I've made? So from the information that I told you, that there is a person called GY, she has 3 family members, 3 close friends, and 3 colleagues or students in the lab, in her lab or in her group or in her workplace. And the edges that I drew out of this was GY to AA, GY to AB, GY to AC, all good. GY to X1, X2, and X3, all good. Similarly, the last 3, all good. But I've forgotten some relationships and I didn't put that. And therefore, my network is incomplete. Can someone tell me what I missed out over here? What did I miss out? Which relationships did I forget? So the relationships that I forgot over here, yes, I do want to have the break that we have in between, because I think everybody needs a break from me. I've been speaking nonstop for a long time, and we will really have a break at the appointed hour, which is 5 minutes. But yes, Yasmin is absolutely correct. The relationship that I missed is the relationship. When I say I have 3 family members, is it that I know all 3 of them and they don't know each other? Obviously AA has to know AB and AB has to know AC, right? So that's a big mistake. So the true network should have more rows. Anyway, let's say I started with the wrong network and I went into Cytoscape. In Cytoscape, we'll teach you how to load the data. You will use the break to install Cytoscape on your own machine, and you will just say file, import, and I will import this file. So this is my network file. Let's call it gy.txt, and you can bring in gy.txt over here. And this is what you will see. GY connected to all 9 people. But then you want to give a different color to it. So this is my text file, how it will look on your machine. And you will take it in, and the problem was I had not put in the relationships with each other. So see, I've put a relationship of AA to AB, AB to AC, X1 to X2. Similarly, my friends know each other and my colleagues know each other also. So then I have these connections. So it's not a fully connected graph yet, because we don't know whether my students know my family or that my best friends know my colleagues. It might not happen, right? You may have distinct networks. And so all you know is that the colleagues know each other, the family knows each other, and the friends know each other. So we have this network that we've got, and we can always change data. So I was able to change the data within Cytoscape, and then we put a visual style to it. So we want to be able to look at the data in a more interesting way. And so that we do by getting here, by changing the, you get a clearer picture, you put node colors, and then you add edge bends. We call it edge bends or bundling of edges, and it all sometimes looks really neat. And then because we gave a different class or category of the relationship to the family versus the office versus the close friends, we can add colors to every kind of relationship. So you see on the left side, you can give a mapping type to each individual. You can say all of my 1 category relationships should be mint, 2 category relationships should be pink, and 3 category relations should be teal. And by giving these colors, you're distinguishing the network visually in a much clearer way. So by doing this, you could do this, in your own kind of work. You could say, okay, my protein interaction data has the flagellar proteome is separate from the centriolar proteome, which is separate from the membrane proteome, which is separate from something else. So you might give different colors to different kinds of relationships, and it makes sense to get a clearer picture by looking at them. Then you start doing the analysis. How would you do the analysis? I talked to you about centralities. I talked to you about so many kinds of different measures you can calculate. So for that, Cytoscape just makes it very simple. You use tools, go into Network Analyzer, and you do any kind of analysis or load the network statistics that you would like to. Similarly, you could have a large number of apps also. And these apps, I've downloaded these apps on my Cytoscape, but you would, when you start, you would have a basic set of apps, not everything. You should go to the web and find what other apps are available. If you don't have time to go to the web, the program itself, Cytoscape itself, will tell you that there are a total of 229 apps. Out of those, you have so many, and if you want more, you can find those. The currently installed ones are here, the updates that you might need are here, the new ones are here, and so on. So what we will do is clustering by MCODE, but not now. We will do clustering after the break. So I'm going to take a break now, and I will be around. If some of you have questions, you can always type them out or ask me, but we will come back in half an hour. You can have a coffee, you can grab yourself a juice or a tea or anything else. I'm going to grab myself some snack, and I'm going to come back in half an hour. You can type out your questions in the chat box. I will be responding in the chat box, but I will generally be mute. So let's just leave the slides here. Shofi, will that be all right for you? Will you please unmute yourself, and maybe we can discuss whether we want to leave the slide on. How do we handle it, or should we put your background, the main background? I think it's good to leave this slide. Is that okay?
You mean your slide?
Yes.
Yes, that would be great.
Okay, if that sounds okay to everybody, great. And if you have questions, as we have mentioned again and again on the chat box, any questions, any feedback, any suggestion to me, just put it in the Discord box. And I'm going to be going to Discord now and reading Discord. So I will leave my slide sharing on, but I'm going to mute. I'm going to check out for a while. I hope you've understood up till now, and I hope you find the rest of the course interesting. Please take some time to install Cytoscape. C2, if you're there, please type out the Cytoscape installation web link on the chat box so that everybody knows what to click and how to install it. It's a very simple installation, one-click installation, and you can, it's not necessary. Those of you who just want to listen to, because I will be showing screenshots of everything, and I will also tell you at every stage why we are doing that. Okay, so if you want to essentially not do it yourself, you don't have to install it, but for gaining the most out of this workshop, I think it will help you if you do take that time to install and think about what I have taught you today. If you have more questions, please ask us about them. So this break, let's take it for grasping some ideas about what we've learned and planning for the next batch of for the after break essence. Okay. All righty then, see you around. Others have also returned. In case you're back in the meeting, in the webinar, would you be able to press the raise hand or yes button on your screen? How would we know? You might be able to raise. Is there a raise hand option? Something like, yeah, I can see raise hands. Good. So if you can raise your hands, I would know that you're there, and we can carry on. Great. I don't see everybody yet, so it looks like, okay. Okay, we are still waiting for a couple of others to come in. Just keep your hands raised till we begin so that I have an idea that about how many of us are back into the webinar from the break. Great. Just to repeat, in case anyone has, in case you're back to the webinar, please join us on the. Please raise your hand in the Zoom chat session. And so I'll know that enough of you are back for us to get the course restarted. We still have a few minutes before, so it's all right. I'm willing to wait. I think in the chat box, if we could just write for everybody to just put a yes in there that they're back, that is also good enough. So I think this also gives me a chance to have an informal kind of chat with everyone to see if there's anything specific that we might have been able to add to what get this done. Did you have other expectations from this webinar or this tutorial, and has it been okay for you up till now? I'll stop sharing my slide because I think what I need is another set of slides. The program itself has been put together by C2, my student, and we've got a separate set of slides for that. Community detection. We will continue with community detection, but there's also, okay, machine learning, AI, no, not this one. Networks, visualization. I think it's the InCoB2020 tutorial. All right, this is the guy. Back to community detection using MCODE. So we can share screen again. Google InCoB. InCoB, right, that one. So there's too many presentations open on my slide right now. On my screen. Okay, great. Back to community detection. This was, of course, there's too many different ways of doing community detection. One of the main things that we're going to be using, and we do use in the lab all the time, is MCODE. MCODE was originally built to find protein complexes, and it was built in Gary Bader's lab. And the paper was a seminal paper in 2004. So what it does is it uses topology to find highly connected regions in a network. And there's a 3-stage process to the algorithm. First, you do weighting. You give a higher score to those nodes whose neighbors are more interconnected. So it's not just about your connectivity to everybody else. It's about how your neighbors are connected to everybody else. So it's in a sense very political. If you are, if you're a node in a political network, your score becomes higher if you're connected to more connected people. So how well do you know people who are important in the whole system gives you a score, and how well do you know people who don't really matter in the system gives you a lower score. So that's one level of weighing that is done by MCODE. The second is molecular complex prediction, starting with the highest weight node, recursively moving out towards other nodes and adding nodes to the complex that are above a certain given threshold. So they try different thresholds and then they decide at certain cutoffs what would be a good idea for keeping some nodes together to say that these form associations that are non-random. So associations that are non-random in terms of protein complexes, it makes a difference, because if 2 or 3 proteins are consistently always found together, let's say during signaling, also during stress, also in a certain anatomical part or organ also, then there is a chance that these 3 are working together or associated with each other over and above random chance. And there is also a chance that these 3 might be physically connected to each other, in a single tight complex. So protein complexes, we have a different series of lectures that we take for protein complexes, whether you have a transient interaction, whether you're a transient complex or you're a more tightly bound complex. If you're a tightly bound complex, then you would spend more time together. If you're a ligand receptor complex, then the ligand will keep falling out and somebody else will take its place, and so on. Or if you're a transient complex, like you're just in a signaling cascade, the kind of interactions and complexes that are made are transient, instantaneous, and then they dissociate and so on. So how would you recognize those as separate from the ones that are more long-term or stable interactions or stable complexes? So all of this is done fantastically in MCODE, and we will do MCODE ourselves today. And there is some post-processing that happens later on. There are some fantastic videos. You should check those videos out. No amount of teaching that I might do today will suffice what MCODE can do for you. The other mechanism of module detection in Cytoscape is which we usually use for module detection for any kind of data set, even in cases where we do clustering and hierarchy, and a lot of machine learning depends on the kind of classification you can do to your data and how you can cluster your data, and whether you use a recursive clustering method or a downwards clustering, k-means kind of clustering method, and so on. Usually what begins is you just start randomly from any 2 or 3 or 4 nodes, and it's best to start with nodes that are more connected. And then you start going outwards from there to find what other nodes are still within the connected area, but not going into another set or vortex of or group of interconnected nodes, for example. And so then you give scores to these sets of nodes which are more connected, and those which are less connected get out towards the peripheral areas. And then these cliques or associations are called modules, and each one of them is detected individually or separately by the program. So this is done by network propagation algorithms, which are of which help you to, you know, get page ranks for Google. When you search something on Google, you wonder why my favorite page is not coming as the first one. When you type InCoB, you know, and I tried this, when you type InCoB2020 on Google, the first hit that you get is not AP Bionet InCoB. What you get is the Frontiers Journals page. And you get the Frontiers Journals page because they've had more hits over the years. They've been there forever. InCoB came in for the last 1 year since they've been, you know, advertising that this is the 2020 conference, log into the conference, do things. There is an InCoB website, but the first page rank is given by Google to Frontiers. So the second one is for the AP Bionet InCoB website itself. And similarly, heat diffusion also uses network propagation algorithms, and the implementation of this algorithm has been done in Cytoscape. So you can use one of the Cytoscape, you can read this paper given over here, which tells you how modules are detected in Cytoscape using this plugin. We will be using MCODE. So you just go to your app manager and download it. Don't do it now. Don't do it now. We will start properly by setting up the workspace and doing everything. But I'm trying to finish off the part that I didn't before I gave you the break. And that would tell you how clustering is done in MCODE and how 3 different clusters. Of course, in this case, you remember the network that we built for GUI, my family and friends and workspace colleagues. So in that case, we had 3 because we started out planning 3 different groups of people. So we expected 3 clusters. And because we expected 3 clusters, finding those 3 clusters would be a verification or validation of the clustering method. So MCODE has perfectly identified these 3 clusters, as you can see over here. On the left, MCODE tells you all 3 clusters. All 3 clusters are of size 3 because there are 3 people involved in each of the 3 clusters. And that gives you an idea that truly this is the right kind of program to use in case you have any such relationships which are not visible because of the large amount of data that you have, or the or the huge interconnectivities, large number of connections that you have in your data. Here, manually, you can see it, but in some cases you can't. And therefore, MCODE lets you choose. You could choose the first, the top cluster, second one, and so on. Importing large data sets can be, we will teach you importing large data sets, which is a part of Cytoscape. But I want to tell you, let's say you have a data set on your computer, then importing. So first thing I've told you is importing data in the form of creating it, literally make a text file and then import it. The second way is to import your input, which could look something like that. This is a gene regulatory information for some set of transcription factors involved in Markantia meristem development, which my collaborators are very interested in. And so we do the same thing of importing it, which we're going to be doing with you. And you first, when you see this huge data, it looks like this. It looks rather scary. There is too much information there, and I just don't make head or tail of it. So how do you then drag down this information, make sense of it, and make it look better and visualizable in a layout that lets you think beyond what the interconnections are and lets you build patterns out of it? This kind of a picture, total chaos, a mess, will never let you see anything. But this is usually the first thing that it kicks back at you when you open Cytoscape. Don't let it scare you, okay? So you can use different kinds of visual styles, which we will use today. You can use different kinds of colors and improve the visualization and see how it looks. Eventually, we try to build from there different kinds of resolutions of the data, separating the data into transcription factors and other genes, and then adding weights. You could add weights or you could keep it unweighted. Those weights might tell you which are the two people which are more interactive, more important in your system, which are less interactive or less important, and so on. Overall, in Cytoscape, you should go to the website. If you've downloaded it by now, I'm assuming everybody has downloaded it and installed it on your machines. But if you haven't, please give me a hand raise and say, okay, I couldn't do it. And maybe then C2 can help you. All right. So it seems that a lot of you are finding it difficult to raise hand. So I'll tell you how to raise hands. To raise your hand, you have to go to the participants. You press the participant button, and okay, I'm a host, so I don't think it's expected of me to raise hands, but you can easily raise your hand by, can somebody tell us, or just type in the chat box how to raise your hand. When I'm usually listening to others, it's so simple for me to raise my hand. And here, so yeah, more. See, when you go to the participants, you have this business called more, and you could do all sorts of things there. So you could then raise your hand or do something or say something. But otherwise, as long as I'm assuming you've got your installation done, I think we're good. If you've not got your installation done, type in a message and one of us will help you. It should have been a simple website download and install, one-click installation. And what I want you to know, what I'm trying to say here is you should go to this whenever, after this webinar, after this course, go to the website and look at the kinds of plugins that are there on Cytoscape. There's a complete database of plugins. And sometimes, when you're a starter, you don't know which is the right plugin out of 300, and there's just no way you're going to be downloading and installing all 300. Choose the ones people are using more often. So the top downloaded ones, the most preferred ones, they'll tell you what are the plugins, and then you can read about that plugin and see what it does. Sometimes you might want to say, okay, I'm not interested in just any plugin. I'm only interested in plugins that allow me to do geo-enrichment analysis. Let's say I'm doing some gene ontology work and I want geo-enrichment. So if you want to understand gene enrichment in your data, you should then look at enrichment kinds of apps, and it will tell you what other apps are there which does that. R we will not do yet. We're going to do Cytoscape first. So back to Cytoscape, and I'm going to present my next slide. Okay. And that is the full tutorial on Is that what it is? Oh, no, that's not what it is. That's the old one. This is the one that I want. Okay. Right. Bear with me. When you have two PowerPoint presentations on screen, I think it becomes a little bit difficult to choose the right one on Zoom. So I'm going to tell Zoom that this is the presentation it needs to open, not the other one. And I think it understood. Right. So for this little tutorial, we're going to be assuming that all of you have a laptop or a workstation or a desktop and that you have Cytoscape installed, okay? We are also going to assume that you know how to double click and open the application on Cytoscape. The entire tutorial has been built on Cytoscape 3.7 version, but as of this morning, just a lucky day, as of this morning, you have version 3.8. So some of you may have installed version 3.8. No harm in installing 3.8, but please remember that this tutorial, because it was made before today morning, so it was made on 3.7. And so if you have 3.7, you can exactly follow the steps that we are doing over here. If you have 3.8, you may still be able to follow the steps, but not exactly. And then I I would not be in a position to immediately tell you go there and do this, because I've myself, when I woke up this morning and I got ready for this workshop, I saw an email that there's a new Cytoscape, but I have not installed it on my own machine either. So I would still say if you have 3.7, that's something that will let you see this. But if you have 3.8, still get into it, look at what we are doing and try to repeat what we've done later on or alongside, and C2 is always there to help you. Okay. So this, of course, has been prepared by my student C2, and she's like totally brilliant. So I'm very happy that she's been able to help me here today and join us for this workshop, leaving her final year research work, but to be with us here today. Thank you, C2. Thank you. Really, it makes a lot of difference. It means a lot to me. So with that, we begin on Cytoscape. I've already told you, Cytoscape is international, it's free, it's by a huge consortium of people who decided to go open access. Now, I'm a huge advocate of open access. Whatever research I do, I have to make it open access. I have to make others be able to do it for free. If I cannot, it hurts me. Similarly, the kind of capacity building and training efforts that we do, it's very important to keep them free. At Cambridge, we run these 2-day, 4-day, 6-day courses, which are chargeable, and Cambridge students and departments are paying for these. But to bring these to India, I had a big difficulty in making them free, because you have to pay for the OTE and you have to pay the entire team and the backup facility at the training facility at Cambridge. So I was looking for scholarships, and now I've got those scholarships. I've been trying very hard for 1 month to get those scholarships, as I told you today. So it makes a difference to make things open source, because I think more people can gain if it's open source. Even this workshop in InCoB, I think it's generally free for everybody who wants to attend it, as long as you're a delegate at InCoB. But the same workshop I have held previously in my Protocols from Home series, and the same workshop I've done with, for different universities in India and abroad. And a lot of people have been able to gain from these. And that is how over different courses, I also learn where I can add more or bring in some more slides to explain a concept better. And that's how things work. Anyway, coming back to Cytoscape, the outline of Cytoscape is that the first step is you import your data. You can import your data or you can create your data, but importing a file could be a file that you've written yourself in text, as I showed you for the GY example, or it could be a text file that you've got, which is your own data, large genetic, genomic, or survey, ecological survey data, or ligand binding data, or cancer patients data, or experimental samples and stress samples data, and so on. So you import that data. The third thing is you don't have any such data, and you don't have the time to write your own data out. You just want to try and dip your fingers into Cytoscape. So there are example data sets for you to use, which we are going to use today for you, so that you don't have to do any background work, but you can immediately try and understand how this works. Then we're going to do data styles. Then we're going to try some layouts for network visualization, and then we will do network analysis and clustering. And finally, we'll take questions, or I will try to give you some tips and tricks by going back to my first presentation. So let's begin. Okay, I'm going to close the video. Okay, perfect. Data import, loading a network. Let's go into File and Import. Click Import. So this is how you go. This is your Cytoscape first window. Go into File, even if it's 3.8 or 3.7. And if you can't do it, just type in the chat box. C2 is there to help you live. I mean, I will not stop the course for one student out of 25 others, but I will be more than happy to have C2 answer each one of your questions so that you're not left behind in this course. I didn't see any chat messages saying you couldn't do the installation. So I'm going with the assumption that everybody's been able to install the software and you've got it with you. So let's do File, let's import. And on the import button, you will have something called network from file, okay? And you can get this from the sample data folder. The sample data folder should be in your Cytoscape downloaded directory. Just go to the sample data folder, and from there you get galfiltered.csv. This is the galactose data, and it's filtered, probably pre-processed. And you get that file and you click OK. Over here, this is how you click OK and get the file. Has everybody been able to do this? Sometimes when we are bringing in our own data, the big issue is that we are not able to decide what is the source and target, which you have to decide if it's a table, what is your source, what is your target, which column is your interaction column. When we brought in the GY data, if you remember, the first column was the source code, that was GY. The second column was was the target code, whether it is AA or B or C, other nodes. That was the third column. The second column was the interaction, 1, 2, or 3. So in my case, I had given you a file where the second column was the interaction, but in this case, the GAL filtered CSV, you have the third column, which is the interaction. And you don't just have the interaction and you don't stop at 3 columns. The data set, GAL filtered CSV file, has more information. The other information it has is something like directionality. So in this network, you have directionality. In this network, you also have a little symbol for each one of the genes and proteins that are being considered here. And you also have a value. A value could be the value of your edges, could be the value of each node, but let's see how that goes. So all I want you to do is to be able to GAL filtered, yes, absolutely. This it should work entirely. Just go to the samples folder and get this file in. Once you've said okay, what you will get is loading its attributes. Now, what has happened is there are different attributes to the same file. Sometimes you upload data about species, and the interaction is just interaction is like species A and species B, and they are interacting in mechanism X. But what are those species? What is X? You need more information about it. And so therefore, what is important is that you should have some node attributes and edge attributes. And sometimes those nodes can tell you that these are transcription factors, other nodes are transporters, some are enzymes, some are regulators, and so on. Edge attributes would mean the edges that we brought in the first stage, which was the stage of when you imported the network. The edges were something like, this is a directed edge, this is an undirected edge, it's a weighted edge, it's got no weight, and if it's a weighted edge, what is the weight of that edge, and so on. So you're going to use file, import, and then you'll get a table from the file, and the node attributes are in the GAL X data CSV itself. The edge attributes are separate. The edge attributes are in something called GAL filtered edge attribute table. So you'll have to do these 2 steps separately. The first step is where you get the node attributes. On the previous slide I had shown you here, you just bring in the table. This is basically node attributes table. So when you do GAL filtered, you're essentially getting the node attributes. And node attributes could mean things like so many other things. You will have a common name to every gene, and you could have an abundance value, you could have an expression value to that. The expression value could vary by different parameters that you've got, and so on. So you just bring it in and say okay. So now you've got the node attributes and you've also got the edge attributes. Okay, press okay in both cases. Now you've got your network. Once you've got your network, you're going to bring in a style. So this is how your network visual will look when you've done no mapping on it. You've just brought in the node attributes file and the edge attributes, and it looks like this. So how do we add mapping? What you do is you just go to network styles. Over here from network, you choose style. Once you've got the style over here, you've got so many different styles that you can use. The styles could be properties. You first go for default style. So in the default style, we are not touching any of the others. In my previous examples, I'd showed you so many non-default styles which have previously been built in Cytoscape already for you, but let's not touch those. Let's do a style ourselves. So click on style. And once you click style, go for default and get here. So I just need to know that everybody's been able to reach this page, and you're looking at the styles now. The properties could be defined by different columns or different mapping mechanisms. Okay. And then what you do is you change the fill color. So nodes, we'll first do node mapping. By doing nodes mapping, what you're doing over here is you're changing the fill color of each one of the nodes. In the previous one, when you first bring in a network, I told you it looks like a blue chaotic mess. So that blue chaotic mess is essentially this. You will see a much darker chunk of data, but you have to zoom into it. Zoom into it by pressing the plus button. By pressing the plus button, once you've gone into the network, you will see individual genes, individual boxes and everything. But these colors and these labels mean nothing to anybody who's reading it. And anyway, you're just in this little part of the network. Actually, it's been zoomed in so much. The network is a huge network like this. And this little piece is what you're looking at. And you have many smaller parts of the network. So it's not a fully connected network, unlike the one that I had shown you from the imported data that we had got. This network is a disconnected network, and it's got some bigger pieces and some smaller pieces. So once you've got the bigger and smaller pieces, you just zoom into one place and then you start applying your styles. The idea about zooming in and then applying styles is good because you see the difference in front of you, even though it's happening to the whole network, but you're watching it happen only to the few slides that you've got in front of you. And so therefore, you're going to be able to add whatever changes you want, and you're going to be able to see it, whether it's interesting enough for you to continue or change. So in this case, what we do is we apply this particular style as shown on this slide. Once again, while you guys do this, I'm going to lock my door and get back in so that nobody disturbs us. All right, so right, so we're trying to do styles. Inside styles, we're still sitting with default. We're going to give our own idea. And in the properties, we are choosing node properties. See, the first thing is you could choose any one of these. You could be looking at nodes, you could be looking at edges, you could be looking at network. Be careful, don't get mixed up. Choose the node first. Once you've chosen the node, you decide what you want to change. Shape, okay? Let's say you want to change the shape of if you want to change the shape of the node, all you have to do is click over here and choose the shape that you want. All right. After that, what you can do is you go for node color, the fill color of the node. For the fill color of the node, so second thing, the first thing you should change is the shape. The shape was square or rectangle earlier. Please make it oval. Once you make it oval, see what happens. This is how you get it. The second thing you should do is change the labels. Earlier, the labels were these meaningless YRF accession IDs. Of course, everything will have an accession ID, but it makes more sense to you only if you put the common name there, right? So you change the name to common, the label to common name. When you click on label, you get multiple options. The options are coming from the same table earlier that you had given to the gal filtered CSV. And in gal filtered CSV, one column was there. You remember I told you about the column called common. The moment you click common, all the names that were the YRF, illegible, unrecognizable codes, accession IDs will now become common names. And so you have common names over here. Okay. So the third thing you want to do is now you've changed the shape, it looks better. You've added the names which look better. Now you want to change the color. You want to change the color based on expression. If something is expressing a lot, the color should be made darker. If something is expressing less, the color should be made lighter. There is a scale to it. So you can go into the fill color, and in the fill color, you choose to have a mapping type which is not discrete. You see, if you put a discrete mapping type, then everybody will have the same color, green, blue, red, whatever you chose. Earlier, it was a discrete mapping type. In the case of labels, we had put the pass-through mapping type. Pass-through mapping type means for every name there is a distinct, for every value in that column there is a distinct value to that cell. So the common name is distinct for each and every different. There's no comparison between them. So if this is PCR1 and this is FW5, they will get exactly that value, which is passed through. So it passes through every name and gives it that name. In the third type of mapping, which is continuous mapping, you're actually asking comparisons. You're basically saying there's a value. It could be between 0 and the lowest value of expression, the highest value expression. Wherever it is, I want that value there. So the color has to be based on a scale, and the scale is continuous. It's not a discrete scale. It's not a pass-through scale. It's a continuous mapping scale. And so once you put a continuous mapping scale, so for everything that you're choosing on the styles page, remember to choose, first of all, click the feature that you want to change, the label or the shape or the fill color. And within that, you will be given the option to choose a mapping type. Should it be continuous? Should it be discrete? Should it be pass-through? After that, once you've decided what it should be, you should choose based on what you want to do that continuous or pass-through mapping. So in the case of fill color, you're going to choose expression. In the case of label, you're going to choose common name. In the case of shape, you're going to straight away click choose on oval, because there are a preset list of shapes. So you can choose diamond, star, whatever you like, but ovals are generally looking good. So we got this done. I hope everybody's been able to map their nodes onto the whole network. If you want to just get out of this box and see how your whole network looks, all you have to do is press 1:1. Do you see this? This little looking glass here. Through this looking glass, if you click it, you get your whole network as a single piece. And if you press plus, which is your zoom in, you can zoom in as much as you like. Or if you want to just zoom out a little bit, but not all the way back to square one, you could just press negative here and it will keep on zooming out for you. So here we have nodes being mapped in 3 different ways. You've added fill color, you've added labels, you've added the shape that you like. You could add many, many other node attributes, but we are not going to be discussing that here, okay? And then we are going to change the edge parameters. Now, how do you change edge parameters? The first thing you do is you stick to styles, but go down to the edge over here. Look at the red arrow. You have to go to the edge. Once you've clicked the edge, you decide the edge width. All edge widths are the same by default. So that means all the edges are essentially binary. They are yes or no, unweighted. But here we've decided to make it a weighted network and give it some edge weights. So the width of the edges is going to be, will it be a discrete kind of mapping, or will it be a pass-through kind of mapping, or will it be a continuous kind of mapping, do you think? When you're giving weights to the edges, the weights to the edges will usually be a continuous kind of mapping, because the value of that weight will be the extent of co-expression between these two people, right? So in this case, because we gave you the edge attributes, we previously already found the a column. We gave you a column in the table called edge attribute 1. So the edge attribute 1 is what you could choose. It could be the co-expression value between those two genes. It could be the extent to which both of them are generally found to be together as often as possible or associated in the cell. It could be anything else that is happening between those two. But for now, you will just choose this edge attribute, which is a continuous edge attribute, and you will give it a continuous mapping. And the moment you choose continuous mapping, all the edges change in their width, and that width value depends upon that attribute which was in that table that you provided to Cytoscape when you began. So now you've got your edge attributes, and now you've got different, you can try different edge attributes. We've chosen one, you might want to try others, but play around with it and see that you can, and then go back again and again. I would suggest you go back to the main network to see how the overall network looks. Are there special clicks or corners or portions of the network which are thicker or thinner or more this from others, or are there not? Okay, so previously, you had a white background to the network. Sometimes we don't want a white background. It's usually good to have, I always say that if you're trying to publish this image, you should keep in all publication quality images, you must have a white background. But if you're making a presentation in the lab, talking to your friends, trying to impress them, I think you should then be choosing more interesting colors that bring out the part of your network that needs to be presented. And in those cases, you might want to change the background. Sometimes if you have white nodes You might want a black background. If you have completely, gray nodes, you might want a very light, a very dark background against them. And so you might want to play around with the colors. So to change the overall network background, you should choose this section of styles, and it's called the network maps. So in the network mapping, you can choose to change the first one, which is the background color. So you can choose the background color, and the background color will become whatever you want it to be. You try doing it yourself. We've chosen gray, but you could choose among the entire spectrum of colors will open in front of you, and you can choose any one of those colors. And so you would have a network mapped to a background color that you choose, with edges whose widths you have decided yourself, and with node sizes and node labels and node shapes that you have chosen yourself. Similarly, you could have chosen edge, how should I say, edge line shape. So if you had gone to the previous one here, remember I told you to just change the edge width. But instead of edge width, you might be wanting to change edge transparency. Quite often in large networks, you have thousands of edges. And even if they are all white colored or gray colored, they will totally obliterate your nodes quite often. And so how do you overcome that? You want to be able to show your edges and your nodes to your supervisor or your friends or your presentation, lab meeting, even to reviewers in a paper. You should change the transparency from here. Transparency values usually go from 0 to 255. And then, of course, you can choose to have, because these are directed, if they are directed, you can choose to have target arrow shapes. The arrow shape may be a filled triangle, an empty triangle, anything else. And you could also have a line type. Line type is very interesting because sometimes you want to separate edges. The edges which are protein-protein interaction edges should all be visibly different. The edges which are co-expression, gene co-expression data edges should be totally different. And the edges which are coming from regulatory data analysis should be in a third different criteria. You could put 3 different colors to the lines, but that would be line color. But you could also put line types. You could make lines straight, you could make them dashed, you could make them dotted. If you remember the example that I gave you in the morning from my lecture, there were these signaling edges which were pink and dashed. There were the protein-protein interaction edges which were green and straight. And there were those other edges which were co-expression edges which were blue and full. So similarly, you can choose the line type here, anything that you choose, and you could also change the labels of the edges. As you will see, this network has no labels to edges. Most networks do not have labels to edges, but sometimes you might want to have a label to your edge. And in that case, you can choose a label color, you can choose a label font, you can choose a specific label to put, everything else. And the color of the edge is always your first thing. In this case, we've put a color of black, which is our default. And we've got a mapping there, which is a multicolor mapping. But you could have chosen for a color to be different based on what category of edge you're looking at. So similarly, each value, the node, the network, the edge, has different mapping features. And for each mapping feature, you have different mapping parameters. And for each mapping parameter, you have different mapping types. And there are 3 types of mapping mechanisms: continuous, discrete, and pass-through. And for each one of these, you can choose what to map, exactly what to map. So this is very, very high resolution work that you're going to be doing. And that makes sense also because you're going to be expressing information about something that is complicated. So now you've got your network visualized. Essentially, we've taught you 3 different types of network mappings, data styles. One is for nodes, one is for edges, one is for the network. What I want you to do is to just do a little bit of what I'm teaching you. The rest you can always do it later because we have given you further reading material, and all of this is absolutely free. It's present in the Cytoscape tutorial sections. Every step is taught to you already, but we want to be here in case, we want you to be able to follow us up all the way to the end of this analysis. And that's more important than getting one part done right. So the next part would be visualization. How would you like to visualize your entire network? So I told you could press that little button which lets you go all the way into the network. Now what we have done, see, this is how the whole network looks. all of the background is white because obviously we tried different background colors. We changed it to gray and then to something else. And ultimately we decided that white is still looking better. Okay, so white is looking better. And then the node shapes, which were all very dark blue earlier and overlapping over each other, they are all becoming small now. This has gone to ovals. You've got the colors, you know just where the highly expressing genes are and just where the non-expressing genes are and so on. But still, this network is just put in front of you in a default layout. How about you want to look at it as a circle? How about you want to look at it as a square grid? How about you want to look at it with a certain group attribute based on expression, based on some edge attribute or node attribute and so on? So let's try a layout. The way the network is visualizable is called a layout, and I showed you, and that's how what tells you what a topology of the network is. Earlier, I had shown you a network that was laid out as a line or a ring or a star. Those were ways of putting the network, laying the network out. So you can choose in Cytoscape so many different ways of putting your, laying out your data. Here we're going to be choosing layout, prefuse, force directed, and just do a default without putting any other character. Press none. What you get is something like this, and then you changed it to edge weighted spring embedded layout. Now you must be wondering, what's an edge weighted spring embedded layout? What's a force directed layout? I've got a full presentation to give you to tell you how each one is different and how each one should be used in what cases and what not. If we have time, we are going to complete this, and I'm going to go back to my presentation to show you those parts. But here it's just to tell you that you can choose any different kind of layout. So for example, this is the attribute circle layout, and you've got all of your nodes laid out on a circle or arranged on a circle and connected the way they were connected earlier. How's this? And they're still connected the way they were connected, but you could choose so many different ways of connecting them. You could choose degree, you could choose a shared name. So all of those would be clubbed together, which have similar degrees. All of those would be clubbed together, which have similar names, or any of that, or their common names, or any of the other features that you incorporated when you brought in the table in the first place. All right. Then you have organic layouts, orthogonal layouts, and Cytoscape has its own set of layouts also. And then, of course, now that we've laid out the network, we've mapped it up with some colors on nodes and edges as well as the background. Let's do some network analysis. In order to do network analysis, we're going to go to tools. We're going to download. There are two tools. I think Network Analyzer is already part of your network. So it comes with the network, with the Cytoscape suite. And so you go to tools, go to Network Analyzer. Within the network analyzer, you can choose to create a subnetwork of some groups of nodes, or you could just do a total network analysis. Let's just do default analyze your network. So let's get this done. Click analyze network. And as soon as you do that, it'll give you an option. The network contains only directed edges. They're not paired, which you could treat as separately or not. You choose, I want to treat the network as a directed network. So then you press OK. You could do the other one. And after this tutorial, I would wish for you to do further exploration yourself. For now, just press treat the network as directed, say OK. And then you will get the results panel. This is your network output. And here it has your in degree, out degree, between centrality, closeness centrality, stress centrality, degree distribution of in degrees, degree distribution of out degrees, average clustering coefficient, shortest path length distribution. You would know what is your shortest path length and so on, neighborhood connectivity, transitivity, everything would be on the results panel. You can download this data and you can put it as a table wherever you want to when you're presenting to your group members, or you could go through each set of results and then see what your network has and how interesting the network is, or how it is different from, if you have two networks, how they're different from each other. So you've performed the entire network analysis. When you do the network analysis, you will get a table panel. It's basically going to tell you so many things about your network, about every node in the network. So you could indeed then the moment you do the network analysis, you get so many values like degree, out degree, closeness centrality. Now they also become attributes to each one of your nodes and edges. And then you can do further analysis of the network based on these attributes. So you then just want to show the hubs. Let's say you just want to show the closed central nodes, and you can choose them, go back to your network, choose a kind of fill color. You say that I want only the nodes that have an edge count of something, or the top between central nodes, and I want them to go red, or I want them to do anything. So you can attribute your network based on network analyzer results also. Now we will do clustering of the network. And when we do clustering of the network, we can do multiple programs, but what we're going to use is MCODE, because I talked to you about MCODE and I gave you the example and we verified it also, how well it worked for us. So go into apps, choose the app manager, choose MCODE, choose install. I'm assuming that you have not got MCODE installed. For instance, in our Cytoscape mode, because we use it all the time, we've got the MCODE running already. After installation, you just go to the, once it is installed, you go to apps, and you don't have to go to the manager anymore because you're not managing the installation any longer. Just go to apps and press MCODE. And then you say, I want to find clusters in the whole network. You could have chosen a small selection of the network, but you're wanting to do it in the whole network. Now, what does it mean to choose the whole network, or to to choose selection? What it means is, when you go back to your network here, let's say this is your network. Now see, there is one big fully connected part of the network, this one. And there are these other smaller bits in the network, these. All of these bits are disconnected with each other, and they are disconnected with the main network also. It doesn't generally make sense for you to do clustering of these little bits, because obviously they will come out as their own distinct clusters. They have nothing to do with other things here. But if you suddenly call these 10 clusters, it is nonsensical, because essentially you don't have enough information connecting these to the rest of the network. So what makes more sense is to choose a selection of the network, the upper part, and then do the true identification of modules or mappings or community detection. It doesn't make sense to detect communities in these little bits and pieces that are hanging like fluff all around you, right? But for now, we're just going to teach you the simple thing to do. And so just choose the default whole network. And then you take a degree cutoff of 2, because less than that makes no sense. Even 2 does not make a lot of sense. If you have a huge network, you should have a larger degree cutoff. But for now, just do that. Out of fluff and haircut, just choose haircut. Basically, it tells you that when you find, I had shown you a picture in my previous slide where you started with a network, white nodes, and you chose one red node, and you got on to give color to nodes around it. You also had a lot of peripheral nodes which were whitish in color. All of that goes away when you give a haircut. So you just get the nodes which are the click of the main network, and not the fluff around it. And so that allows you to find more, refine the network identification and get exclusive clusters. And then you press analyze current network. So all the values are default. You analyze the network. The moment you click analyze, within a second, it has analyzed the entire network and found for you 7 clusters. These 7 clusters are made up of, first of all, they're given a rank. So those ranks are over here. And these are not the ranks. You have basically the number of nodes and edges is the first thing that it tells you. And among the nodes and edges, yes, these are ranks. So you have ranks, and then you have the nodes and edges. So if two clusters have the same rank, they will be placed based on the number of nodes in them. So even though this cluster has the same rank, but it has fewer number of nodes, therefore it is placed after the other cluster, which is the greater number of nodes. Now my question to you, so your question would be, where is this first cluster? My network is so huge with hundreds of nodes. Where is this cluster with 4 nodes? So you click on this. The moment you click on this, all of the nodes get colored, the ones that belong to that network. So this is where you began. You did a network analysis and you found that you have 7 clusters. And then you suddenly found that your gene of interest is in the second cluster. How do you know your gene of interest is in the second cluster? Here on the table, every cluster and its what do you say, composition is given. And so you've chosen the second cluster, and the moment you choose the second cluster, all 5 nodes light up and they are here. So this is how you identify the clusters. But go back. I have an exercise here. The exercise is, I want you to, now that you have these 5 nodes colored, I want you to color the edges that connect these nodes also. Can somebody do that? Can you think, based on what I've taught you about network mapping and edges, nodes, and full network, Sometimes when you show a network to somebody and you say these are the 4 in the same cluster, it doesn't make sense. I want all of the other edges to become transparent, and just these the edges that are connecting these 4 nodes or 5 nodes, they should remain visible. So you don't have to make everything else 0. You can still leave them be, but just the whole subnetwork should come out individually as it is. You don't have to do it now, but think about it as an exercise. Maybe you can do it alongside and then tell me whether you could get it done or not. Also, even after the workshop, you're welcome to try it out. If you can't do it, write on the Discord chat box and I'll tell you how to do it. I'll tell you the trick to get the edges colored also. It's very interesting. Okay, now we will do clustering with another app. It's called Cluster Maker. Once again, you go to Apps, App Manager, get the Cluster Maker, install it. After the installation, go to the apps once again. And you would just straight away to the apps, you would find Cluster Maker is sitting there already because you have previously installed it. From Cluster Maker, now Cluster Maker can do a lot of things for you. It can do clustering in so many ways. I told you MCODE is just one kind of clustering. MCL is another kind of clustering. Many other methods are there, you know, and you should be able to use any of them or compare your clustering, hierarchical clustering, k-means clustering. When we do machine learning, we are always teaching each one of these types of clustering methods and individually hand-holding students till they understand each clustering method is separate from each other. But for now, for you, it's all been done already. So you can choose one of these. Let's choose hierarchical clustering. In hierarchical clustering, a smaller window will open up for you. Let's just choose the all default parameters. We're going to do simple Euclidean distance. That means only the basic topology is going to be used currently and nothing else. So you're going to be deselecting the option of only using selected nodes or edge because you want to use everything. And then you're going to be clustering attributes as well as the nodes. And then you're going to be ignoring nodes and edges which have no data. That's very good because you don't need to bring in clustering values where you don't get anything. So node attributes based on which clustering is to be done, you can choose over here. You create groups and select the option show tree when complete. So it might take some time depending upon the size of the network that you're working with. If we had been doing with the small GY network, it would have taken nanoseconds to get here. But now because we are using the GAL filtered network, it will take some time. So you press OK and then you see what you get. You get this entire heat map, which basically tells you what kind of clusters are there in the data based on the attribute that you had chosen, which was the expression values, GAL4 expression values in these 3 columns. So you chose the node attributes for clusters as all 3 of these, but you did not choose any edge attributes. So you can sometimes do clustering based on edge attributes, or you could do clustering based on node attributes. Here we are doing the simple node-based clustering. And once you get it, you get a total heat map. You just shift drag to draw a box and see the results. So now all of these essentially are all of your genes in the entire network, but you're not interested in them all. You're just interested in the ones that are visible as green over here. So you just shift and drag from one corner to here. The moment you do a shift and drag, something like this comes up, and then this little box, you see only those nodes on the network. And then what you can do is, because we are on a Mac, usually everything we do shift, otherwise it's right click. You do shift click to pick individual columns. You might not be interested in all the columns here, right? Or if you had chosen this one, you might only be interested in the green columns, right? So you can select individual columns and you can also select individual rows, and you can change the settings to change the color scheme. If this looks too dark to you, or if you're not happy with the red, green, the whole world uses red and green, you want to go for blue and light blue, you're more than welcome. You can adjust the color scheme and the contrast that you find over here by going into settings and changing whatever you like to change. Most essentially, when you find, the scale is always visible over here. You should be careful about what scale you're looking at. And when you've picked these guys, once you've selected only a few genes, those are the only genes that are going to be visible on your table. So you can make sure that you have the right kind of genes that you're interested in. The other way is you just tell Cytoscape that my gene of interest is the PVS1 or CLN3. And so then it will already tell you where CLN3 is in this whole entire heat map, and that area is going to be lit up for you. Now, how do you map a color onto the network? So now that you have this GAL1 color and you want to be able to map that color onto the network, you go into, again, map the colors onto network. Choose it here. The moment you click it, you get options of the 3 colors or the 3 columns that you had chosen earlier for clustering. And then you say, create a map based on this. So that there'll be an alternate map that you will get based on which all of the node colors are going to be given based on just this experiment. So then just based on this, all of your colors have changed over here. So you could choose this, but you could have chosen the other one, which you had chosen earlier, and you would have given it. Sometimes it makes more sense to see it like this. Sometimes it makes more sense to see it in the other way. But whatever you do, I think it's most important to, which I keep telling my own students also, you have to play and play some more. You have to constantly use your data and constantly work on it and try different things with it and fail 100 times before you succeed once. And that would make more sense. So don't ever just put your network onto Cytoscape, and whatever is thrown back to you by Cytoscape, don't take it to your supervisor. It will be the worst thing you can do. Please spend some time and have some regard for the time that your supervisor has or your presentation audience might have. Spend some time, build good networks, and see what makes more sense and what is more sensible, and then you take it on to show to others or put into your presentation. Now, would you like to animate your network? You could animate your network based on the different time. If it's a time series data, you might be able to see how differently at each time the genes change in their color or change in their values. And sorry. So you could do animation also. I think Cito has not been able to put that video because that would be an active video. So what we have then is most essentially a very simple series of what we just did just now was to build your own network and go into it. What I'm going to do is I'm going to take you back to my previous, presentation, so that I can give you some tips and tricks and tell you about the different kinds of network features that we had earlier. The layouts that I had taught you, those layouts you might want to understand a little bit better. And so here it is. We're back to my original slide. I hope everybody can see it now. And we did analysis. We tried Cytoscape ourselves today in a little step-by-step tutorial. And I hope some of you, at least even if one of you has been able to understand how you can use existing data and visualize it or convert tabular data to a network, I think I've achieved my objective. So that's the best I had set out to achieve. So this is R, but we have very little time. So I want to skip R, and I want to be able to tell you what things you can do in Cytoscape only. So I'm going to skip over the entire tutorial that I had prepared you for R. Please try it out yourself, in case many of you did tell me in the morning that you've been using R previously. R, iGraph and SNA have fantastic hands-on tutorials where they tell you how to make a network from scratch or how to import data sets and various kinds of things you can do over there. Even for Cytoscape, you have all of this online. You should be able to try it yourself. So there are some kinds of so network aesthetics and tips for visualization is my last section for today's tutorial session, today's workshop. And in that, I would like you to know the kind of challenges you have. When you begin with network analysis, the biggest challenge you get is that your networks can sometimes be very large, and therefore you will have display issues. Depending on whether you're using a laptop or your workstation or a big server, you may have display issues because larger networks require more graphics capabilities in your workstation. The second thing with larger networks is you sometimes have no idea what you're looking for because there's so much to look at. You just don't know how to boil into the important pieces and where the patterns are. And therefore, it's extremely important to establish collaboration between computer scientists and biologists. Of course, we are having more and more research scientists coming in who are computer scientists but wanting to do biology. At Cambridge, indeed, we have far more cross-disciplinary scientists than we have here in India, but the numbers are changing. We have PhD students from biology who are going in, especially working in the IITs with computer scientists, and we have computer science students who are coming into, you know, NIPGR, for example, to want to do their trainings and so on. I had a physics student with me a couple of years back, and we did some very interesting work. So similarly, there's lots of kinds of things that you can do. I think there's an urgent need to bridge the divide, but this is not what I wanted to teach you. Yeah, effective visualization strategies is what you need to learn. For example, how you can provide readable labels and captions. If you compare these two pictures here, sometimes the labels are so small they make no sense. Labels have to be big enough. Sometimes students leave so much white space on the screen and the labels are just not readable. So make them readable, make them nice, choose the right level of detail. Don't put too much information that will not make sense. Use color responsibly. The left side picture over here has just wasted color. You don't need to get married right there with all this shoshabazi. You can just have a more laid back, you know, you don't have to have a celebration of color in that moment. It's still a research work. You can use shades of the same color and that makes more sense. And then you should use multiple figures rather than a single figure. You can choose the same big network and different parts of it in smaller figures. And then you should also never do unjustified 3D. Now networks can be converted into 3D and Cytoscape has some fantastic 3D plugins, but I would suggest please don't unjustified unnecessarily put 3D when you don't need it, and 2D is good enough for you to show your data. What counts in your network is the use of color. Internet nodes could be colored by out degree, edges could be colored by degree of the endpoints, and you could use meaningful coordinates like polar coordinates, etc. You could use different sizes. You could color the nodes size by degree. I had shown you the transitivity colors and the closeness centrality colors in my initial slides, and there all the nodes were colored by their degrees. And that's how you could tell me that the red node is more central, orange node is red, less central, and so on. The other things that matter are node shape and edge thickness. We have seen in the little example that we took today how you can change node shape and how you can change node thickness also. And I hope that has made sense to some of you. And of course, network aesthetics, the most important part of network aesthetics is when you have too many nodes, do not cross the edges. When you cross edges, it makes a mess. So you should minimize edge crossings. That's why this kind of a network visualization would be better than this, okay? Similarly, don't change edge lengths too much unless and until you need it. If this is how your network looks, you don't have to drag one node away from each other manually, you know, to show that these three are one group and this is better. Similarly, never allow nodes to overlap with edges that are not incident on them. Here, you have these 5 nodes and this is how they are connected. But if you manually drag and pull this node onto this one, it will become like this. It might look cool to you, but it's giving a wrong impression to somebody else that these 3 nodes are somehow interconnected, which they are not, right? So don't make such mistakes. These are aesthetic pieces that are very important. Similarly, you could represent nodes and shapes, nodes and edges. As I told you, we took the example. You could do thickness, labels, colors, shapes of edges. Similarly, color, size, shapes, nodes, and locations of nodes. Okay. Now the layouts. Layouts are extremely important. We saw how we can give a circular layout, a tree layout, a planar layout, you know, to the network today. What layouts are doing is they are conveying the relationship between the nodes. There are many algorithms, as you saw, which need to be tuned to fit your needs. And the one single thing that I say is try them all. Do not assume that one is good for you. In a circular layout, all the nodes get put along a circle and all the edges between them are drawn in the center of that circle. The big advantage of the circular layout is that circular coordinates can sometimes represent a property of data. Remember when we were clicking the degree, or we were clicking closeness centrality, or you were clicking the names of common names? You could put these nodes in a certain order, that everything which is having a high degree comes together and everything which is lesser degree comes to the other side. Or you could say, you know, the age of all people would be the reason for which you want the circular layouts to be put. The best advantage of circular layout is it's immediate, it's quick, it's the fastest of all layouts. But there are many disadvantages to circular layouts. Number one, if you have a large network like the one you're seeing in front of me, it is very difficult to interpret or make any sense of this big earth-like box, right? What do you do with it? And also, circular layouts lead you to have many overlapping edges, which is not good. Also, there are many long edges, even though this node and this node, are could be connected to each other, such a short one. If this node were kept here, this length would be very long. So the length of the edges makes no difference, but long edges become very complicated to understand. And then of course, connected nodes need not be close together. So even if two nodes are connected, let's say these two nodes are not connected, they are still sitting next to each other, giving you the wrong impression that they are connected. But this is connected to this guy and they are completely on the diameter. So they are so far from each other and these are so close to each other, even though they are not connected, because you've put them together based on node attributes. And so therefore connectivity, you have no idea. And that's a loss of information. Similarly, the clusters in such a mechanism are very hard to identify, especially if you give a certain attribute that I want the circle to be based on degree. You have forced a visualization which may not actually be there. So the clusters which are there are just not possible to recognize. So try other layouts also. Among the other layouts, one is a radial layout. Radial layout is very interesting because it starts with one node and it starts to draw all the other nodes in circular layers based on the steps taken to reach them, the shortest path mechanism. Didn't I tell you earlier that you can go from one node to every other node in a certain number of steps, and that number of steps decides the number of paths that are there, the length of the shortest path. And so radial layout allows you to read the shortest path length in one go. For this network, for example, the shortest path length is step 1, 2, 3, 4, 5, and 6. So it's a 6 degrees network. So similarly, radial is therefore interesting. Then spring embedded. Spring embedded has two parts, and we had done the spring embedding together on the example that we took today in the tutorial. The two parts of spring embedding are, number one, there is a force that quantifies the quality of the drawing. And second is an optimization algorithm that computes a network configuration locally optimal with respect to this algorithm. So when you begin with each network, the force on each vertex is 0. But the final layout will depend upon the starting positions. So therefore, there is some level of iterativeness and randomness in this, which helps the algorithm find the global minimum of the best way of laying out something. So this kind of spring embedding layout, the advantage is that you see relationships and clusters that you don't usually see in other cases. But the disadvantage, what do you think is the disadvantage? Simulated annealing and random positions. Imagine the number of times this is going to go running again and again. Similarly, the spring embedded can be in two types. One is Fruitman and Reingold. The Fruitman Reingold method, which you did try today, it roughly corresponds to the electrostatic attraction between the connected nodes. So if two nodes are connected or 3 or 4 nodes are more connected to each other, they'll be closer. If others are broken pieces or disconnected pieces, they'll be thrown a little bit farther away. But this really lets you see the clicks or the associations in very large networks. So this is an iterative optimization algorithm where at every step, every node reacts to the pulls and pushes of the springs that tie it to all of the other nodes. So every edge is considered as a spring. And if there are 3 nodes like this, these 3, they are pulling this node towards themselves, whereas this node is not able to pull it closer to itself because these 2 are bigger than this one. But this node is entirely to the rest of the cluster because it's being pulled by at least 6 or 7 different springs to itself. So this is the Fruitman-Reingold method of layouts. And the other one of spring embedding is Kamada-Kawai. Kamada-Kawai is interesting because it is a kind of spring embedding, but still it has a circular idea to it. So all nodes are connected by strings with a resting length which is proportional to the length of the shortest path between them. And the big issue is that you need to calculate all pairs of shortest paths for every single pair of nodes, which is the shortest path. And that will go on iteratively optimizing itself. The big advantage of Kamada-Kawai, and you should try Kamada-Kawai, is that on edge-weighted graphs, it gives you the best kinds of clusters. The true biological meaning comes out in the Kamada-Kawai. But the disadvantage of Kamada-Kawai is that it can take so long that you start to wonder whether you're going to die of old age before this thing will ever complete. But if you do want to try Kamada-Kawai, and I suggest you should try Kamada-Kawai, you should then use a bigger desktop or go to your labs and get a remote workstation or access a remote workstation which is stronger and can give you the Kamada-Kawai on a larger number of group nodes. Okay. Then the other one is the graph embedding layout called the GEM layout. In the GEM layout, what you have is you have an embedding algorithm with speed and layout optimizations, which are significantly faster than the Kamada Kawai, also faster than the Frutum and Rheingold. So these are very useful for large networks, because quite often we'll work with protein interaction networks are not so large, but the gene expression networks or gene networks are much larger. And you have certain, in Cytoscape you can do it in one way, but there's another program called Guess, where you can lay out up to 10,000 node graphs depending upon the edge density, and you can do it in seconds. So that's another program that you might want to look at some point of time, especially if you have very large networks. Okay. The big thing about visualizing large networks, which you should remember, is that first of all, if possible, please remove redundancy. There's no need to keep every single thing under the sun unless it makes sense to you. So introduce thresholds. So if you're doing a bibliography network, for example, use only those papers which have been written by authors who have at least 5 or 6 or 10 or whatever number you want to put, some cutoff papers. If you want to use edges with gene co-expressions with a certain weight which is above 5 or not. Or you could use degrees, only nodes which have a degree of Z and so on. So you can remove everything else. The other thing you could do is you could show minimum spanning trees, or you could use Pathfinder for network scaling, or you could collapse your nodes into clusters. And we do that in WGCNA, which possibly if we do another course with you, we'll teach you how to do that with genomics data sets. The other big aesthetic issue is cool looking visualizations are not always the most informative. So over here, this is far more cool looking, but doesn't have that kind of information about the layers in a food web. Here you see the different levels in the food web structure and the pyramid. So this is far more sensible than what you get here. And food web networks are made by this specific program, or I would say, the software that is used here. You should look at, I used to use it very often when I was doing my PhD, but that was like 15 years ago. So I can't remember the name of this one. It was very good for food web analysis, but please do take a look at the ecological monographs paper that this has come from. And then of course, spatial interpretations can be very important. So this is the mouse brain connectivity data, and both are the same figures, and it's visualized in the COSE program here using a spring embedded layout. And the thing is, the left image, and see the difference between the left and right images. The left image uses the connectivity strength as the driving force for the layout, and it poses strongly connected nodes closely together. But at the same time, what it has forgotten is the spatial context of the network. In a mouse brain, there is so much known perfectly about where every gene is, where every position is, that the layout on the right side is taking into account the spatial relation of the brain regions. Different regions of the mouse brain have been very well characterized. And so you can generate automatically a flattened kind of mouse brain representation seen from above, which allows you to see that the nodes which are in this part of the brain are all having more similarity to each other than the nodes in this part of the brain, which are a little bit different. And this brings in more sense or a better pattern than you saw over here, right? So you should know how to visualize your network under which circumstance, and when clusters would be good and when spatial interpretations might be more important. This is our floral transition network. Here we've separated the network by intercellular locations, like the nucleus in the center, and then we go to the plastids, chloroplast, endoplasmic reticulum, Golgi apparatus, plasma membrane, all the way out to the extracellular and cell wall areas. And that's how we see that one gene which we started with reaches out to so much in the floral transition pathway. Also, there is this little subset of transcription factors which are only in the nucleus, which are all making some sense or interacting with the making a little clique in here, which allow you to then expand out and and try to understand how floral transition is connected to other pathways and so on. So spatial arrangement of genes or spatial arrangement of nodes can sometimes be more important than functional arrangement of nodes. Okay. Other tips of network visualization, all right, if you want to remember just one, the success of visualization is based on a deep knowledge and care about the substance, quality, and relevance of the context. So one rule, take home message is know your network. Don't you not know your network, because that doesn't help. And the 5 principles in the theory of graphic display are: above all else, show the data, not the design. Maximize the data ink ratio. Don't leave big white spaces, this means, okay? Second, erase the non-data ink. Sometimes students have a tendency to write this, that, put too many annotations. Just get rid of any ink that is non-data. within reason. And then you erase the redundant ink. Sometimes you end up saying things twice. You can say things twice and thrice to emphasize them, but no need to write them so many times, okay? And in the end, always, always revise your data and edit your data. Okay, with that, I would like to thank everyone for listening to today's workshop. I especially thank C2. She's my final year student. She's been here and she's been able to take questions from most of you because I haven't been able to look at the chat box so often. I will do now afterwards, but of course, that's the main thing. We still have a few minutes, Shofi. Should we do some, should we have some questions, or do you think the Discord would be okay? Should we disperse? How should we handle this?
Yes, we still have like 18 minutes to go. So I think
So if there are any questions, I'm happy to
yes.
take. Yeah.
There is a question in the chat box, and also I don't think we have any questions from
Okay.
Discord.
What do you think about data reduction in network analysis? How far could we reduce data dimensionality in the data, or should we not do that at all? So that's a good question. The first one is about data reduction. When I say data reduction in the analysis, this can be looked at in two ways. One is, as I was saying, we could bring in some how should I put this? We could bring in some cutoffs, right? So when you're looking at gene expression data, for example, quite often you will find that two genes are co-expressing with each other, and you could have the co-expression scale from 0 to 1, in fact from -1 to +1, because there is negative co-expression also. So in those cases, you don't want, because there are thousands of genes, and if you consider 20, let's say 20,000 genes in a regular genome, 20,000 cross 20,000 would be a hugely colossal number. And it's best to not look at all of that data. To what extent within reason can you get rid of information that is not required, or information that is just background noise. So if you have a -1 to 1 range of co-expression in your co-expression network, I would say get rid of everything that is worse than positive 0.8 and negative 0.8. So any genes, any pairs of genes that are co-expressing with each other at a co-expression value of 0.8 or more should be retained, and everything that is -8 or less should also be retained. But between -8 to +8, you just don't need to keep that data, just chuck it all out completely. In some of our cases, what we find is even at the 0.8 cutoff, we find a huge number of interactions. So there would still be false positives. And especially when you're doing a computational research, you need to get rid of false positives, which is far more important than getting the correct true positives. So getting rid of false positives would mean that you should possibly then take a much higher stringency of cutoffs. Maybe you could take 0.9 if you wanted, and bring in significance values like p-values, which we do usually on a regular basis, to remove this. Again, in a bibliography network, I would say you should remove everything that is from single connection. So if an author has just made one paper ever, unless there's a citation, so unless that single paper has changed everything, like when Einstein first wrote his first paper on the general theory of relativity, I mean, then he wrote 5 papers after that, but before that he had nothing. And all his papers, if you looked at the first paper written by Einstein, he didn't give a single reference. That means he had never read anybody else's work. He just sat and thought and then wrote the theory of relativity, and that was incredible. So that single paper would have made all the difference in the world, but most people are not like that. And so therefore, I think it makes more sense to consider authors who have written a certain number of papers and remove that below that. But again, you have to know your network. You know your network, you're looking at a certain number of people, and you know that in this network, the important papers include those which have been written just by one author only once. In that case, you would need to change your cutoff based on your context. Okay, so that's one way of reducing data dimensionality. The other way of reducing data dimensionality, see, reducing data is one thing, reducing data dimensionality is one thing. So reducing data is by introducing cutoffs, and reducing data dimensionality is by essentially overlapping data. When you remember the first network picture that I showed you, the cell biology network, in that you saw that you had the same 5 nodes, or 6 nodes, and within that you could see the protein-protein interaction also, gene expression also, signaling also. So you're putting layers of information, you're superimposing data over each other. Rather than seeing 5 different networks or 5 different tables, you're seeing a single network, and on that single network you have all the pieces of data. So you've very nicely reduced your data dimensionality. Mathematically, reducing data dimensionality is an idea of doing clustering. It's about putting things together which are very similar to each other. Okay, I hope that helps. Good. The next question is, can we predict the PPI network that affected by bioactive compounds where this compound has inhibition action to the specific protein target? Is it possible to analyze its PPI? Okay, this question is vivid. Dre, I've not been able to understand your question. Can you rewrite or rephrase the question one more time while I take other questions? Not a problem. What do you think about data reduction in network analysis and how far we could? I have handled this. Okay, good. So the next question is by Wong. Should we use Cytoscape to generate networks and adjust manually? Yes, definitely. You should generate the network, and you should sometimes adjust it manually. Very recently, we built regulatory networks of plant memory. So in that, what we found was we found 5 interesting clusters, and these clusters were connecting epigenetic memory to protein-related memory. But the clusters, one edge of the cluster was overlapping on the other. So we manually pulled out and separated the 2 clusters so that the relationships, the edges and the nodes could be visualized. Plus, we also had the names, the common names of these nodes on each one of those cluster names of the genes on each of the nodes. And quite often, the way the default layout algorithm works, it just does try to keep the node labels separate from each other, but beyond a point, you can't do it, right? So manually, you will have to improve it. Yes, indeed. So you must always look at your network hundreds of times if you have to, and you should also adjust it manually wherever required, just as you do in a multiple alignment. The next question is, should we use Cytoscape to generate networks and adjust manually? Oh, yeah, right. Absolutely. That's what I've answered just now. So I didn't get the second question from Prasanthi. So I'm going to try and understand what I made from your question, and I'll try to answer it based on my understanding. So the question is, can we predict the PPI based on inhibition action? So you can't predict PPI based on inhibition, but you can predict PPI based on HPLC data, let's say, or mass spectrometry data, let's say. Even then, predicting protein-protein interactions is a very, very, very difficult mechanism. What you can do is you can say protein presence-absence data, also protein location data you can bring in. And if in multiple sets of location data sets, 2 proteins are constantly found together, and then when you overlay it with co-expression data, or expression data, and you find that both of these proteins are expressing at the same time in the same location in the cell, then you might be able to say that I can predict PPI. But generally, PPI predictions are not very easy. So inhibition action to the specific protein target. Again, is it possible to analyze PP? Again, it's very difficult for me to understand this. What I'm trying to get is, you have something like inhibition action. Ugh. So I'm assuming that you have a protein and target, and you have an inhibitory interaction going on with that specific protein, and you are aware of the residues that are involved in the inhibition, or maybe you have the structural information. If you have the residues lining the binding cavity and the residues involved in inhibition, and you build a network of, let's say, hydrogen bonds within, or let's say you build a network of salt bridges, then you might be able to predict how to break that apart. But you can analyze the PPI, but still you can't predict it. So we do a lot of intramolecular networks of proteins, and they are based on protein ligand interaction, but it requires you to have interaction information, detailed interaction information. I hope that answers your question. I don't see any more questions, Shofi.
Right, and I actually just unmuted Vivitri. Maybe she wanted to ask the question herself.
Okay, thank you. I actually already rewrite it in the chat box. Well, probably I can tell you directly. For example, if I have estrogen receptor alpha, which is inhibited by a phytoestrogen compound, can we predict the effect this compound to other protein via protein-protein interaction that connected to the ER alpha? Is it possible?
Just a minute, I'm going to charge my machine before it dies out on me. Great, so now my machine is charging. All right, so what I understand from you is you have a protein-protein interaction, one structure interaction, and based on that, you want to predict if other proteins are interacting or not. The answer is yes, only to some extent. So let's say your receptor and ligand shows you that a very important feature of your interaction there is two residues which are interacting with the ligand. If these two residues are present in other proteins at the same position when you do a modeling, then you can say that possibly the mechanism that is required for inhibition is present in those other proteins. But you cannot just make the assumption based on these two alone. What you need is more information, potentially more information on structure, potentially more information of how of these two hybrid. For example, if you have a whole genome Y2H data set with you, then you might be able to make some predictions or remove some false positives from your structure-based predictions. Does that help?
Okay, thank you.
If there are no more questions, what do you say? Should we
Sorry,
close this meeting and get on
more.
Discord? I'm happy for you to come and interact with me personally on Discord, or write to me if you want to, or follow me on Twitter and there ask me anywhere you like, not a problem.
There's still one question.
Is there another question? Where's the chat box? Is this? Can you explain a bit more about loading different kinds of data inside Cytoscape? For example, matrix-based correlations. Oh yes, absolutely. So, so let's say you're working in R, and let's say you've got 17,000 genes, and let's say you have the expression information of these 17,000 genes across 10 different experiments, or let's say 20 different experiments. It's a time series data. So you've got a 17,000 cross 20,000 matrix of expression values. What you have to do is take every pair and find the correlation between each gene with each pair of genes across the entire span or spectrum of the 20 experiments that you did. Then what would you have? You would have a 20,000 cross 20,000 matrix, because you would know which two genes are expressing to what level with respect to each other. So from this 20,000 cross 20,000 matrix now, so from a 20,000 cross 17 matrix, you've now reached a 20,000 cross 20,000 matrix. So this matrix is your co-expression matrix. In this matrix, you decide immediately to put a cutoff because you don't want to see everything like we just discussed. You want to remove the fluff. So you remove every cell which has a value of co-expression correlation coefficient as less than 0.8 or more than the negative 0.8 to become 0. Immediately, you've removed everything that doesn't make sense. So all of those cells become 0 for you, and that is one way you have reduced the data or removed the unnecessary data. Only the rest of the cells have values. These rest of the cells have certain values which are called correlation coefficients. But let's say you want to keep it simple and build just an undirected network. So you can convert all of these to the value of 1. So you have a yes or no in case of an unweighted undirected network. Now you have a matrix of 20,000 cross 20,000, but many of these cells are 0 and some of these cells are 1, exactly like the adjacency matrix that I showed you. So from the original data matrix, which is your gene expression matrix, you got a gene co-expression matrix, and then from the co-expression matrix, you built an adjacency matrix. Now that you have an adjacency matrix, you just decide that anybody who's got a 1 is your network, is an edge on your network, and anybody who's got a 0 is not an edge on your network. So you take that adjacency matrix with its rows and its columns, and you convert it to an edge list. So when I say you convert it to an edge list, you're basically saying row by row information of which two pairs are connected to each other. In R, there are lots of scripts that will do it for you directly, automatically, but you could also download that entire matrix and write a simple Perl script to get yourself the pairs. Once you've got the pairs, it's essentially a comma separated file with edge lists with you. And these edge lists have come from the adjacency data. So these edge lists can then be incorporated into Cytoscape exactly the way we did for the GUI file. The other alternative is you stick to the adjacency matrix and you stay in R, and in R you build the R networks, which is again a complete world of fabulously making very good networks. So you could convert the adjacency matrix to a graph in R using the R-igraph package. You could also do it in this SNA package. You could also use the, in R you have this Shiny, so Graph Shiny, a new package called Graph Shiny, which does really neat smoothing layouts for you. So that's there also. But if you want to just, based on your question, if you just want to bring it into Cytoscape, you convert it to an edge list. Once you have the edge list, you download the, or write out the edge list onto a CSV file, upload the CSV file into Cytoscape, and there you are, it's done. Anything else?
I think there is a question from Awa Filbert Liko. So may you
Okay.
explain about loading the obtained network from string into Cytoscape?
All right, very interesting. The string generally gives you PPI networks, and the string will let you choose whether you want to use only characterized protein interactions, or would you like to go in for things like predicted protein interactions also, based on n numbers of different options that you have. So first thing you should do in string is to remove the cutoff parameter, because by default it only gives you 50 outputs. So use something like 500 or 400. So you get a large number of potential hits, and then you get a score for them. So you will have a column where you will have your interacting partners, the first interacting partner, the second interacting partner, and they will also, string is really good. So string will also tell you common names in the third column or fourth column. String will tell you a certain score, which will be the score of that interaction if it is really there. And the fifth column would be, string will tell you what kind of an interaction it is, on what basis or what evidence it built that from. So string uses up to 5 different evidences, or even more, I think, and each one of these evidences can become a separate column on your data. So then you download the string data and you keep all of these columns intact. So you will have more than 7 or 10 columns in your data set, and each row will be an independent protein-protein interaction. So then you import it into Cytoscape just the way I showed you with the large data set. Anything else? I hope that helps. Try it. You should try it. Okay, there is another question.
Yeah, there's another question from Yajan. Thank you so much for the great lecture, but if you allow me, what course, software, or any other resources would you recommend for a beginner in bioinformatics?
If you're a beginner in bioinformatics, the first thing you should do is to pick up R, and R has many basic basal tutorials online, freely available. The second thing you should do is to pick up Cytoscape, which we have done today. So I think as long as you've looked at Cytoscape and R, you're pretty good. A lot of our students, they tend to think that doing a lot of structure-based studies will be useful. I don't think you should be learning Autodoc and stuff like molecular modeling, because very few people depend upon that these days, and a lot of wrong information is being spread among students that if you just do a docking, you can find out how two people are interacting. Protein-protein interactions are far more complex, and it would be much more useful, even if you want to study protein-protein interaction, to just stay away from docking and molecular modeling, but to better go in for things like trying to understand the intramolecular hydrogen bonding network, and that would come out from the interface residues, and the interface residues can be accessed by programs like NAccess or PDB-Sum, and looking at those things rather than predicting too much, which has a lot of errors. And so therefore, I think it makes more sense. More computational biologists should be using experimental data for making the predictions rather than modeled data. So I am more for using actual real data rather than modeling based data. And so therefore, as long as you're doing R and Cytoscape, I think you're in very good position to start bioinformatics, and then go on to genomics and use R bioconductor. But using R bioconductor is not the first step, should never be the first step. Even if you spend 20 minutes doing R every day, I think you'll be an expert by the end of 2 weeks, and you would be so desperate to do more, it'll be difficult to drag you away from R. So pick up R. If you're a lady, join us on the women's workshop for data science next week. So first you pick up R, basic R, as we are going to teach you next month. But after that, you pick up data manipulation, data visualization, and then you do genomics through bioconductor and networks. Of course, there are so many programs in network, are plugins in R for doing network analysis, but Cytoscape is easier because for a starter, you don't have to write any code. You just have to click things and something happens. So I prefer that as a beginner, you should play with GUI based interfaces. But if you're wishing to put your, dip your fingers deeper, you should probably then be looking at coding based things like R and RStudio. Don't use R, use RStudio. Okay, great.
Thank you. For there's still one question from Sona.
Yes.
When is the workshop and how to register it?
Okay, I'm going to put the link to the workshop brochure into the Zoom chat box. I could have done into the other one, but this is the workshop brochure. Please take a look at the workshop brochure, see what we are teaching you, and if you're interested, please attend it or apply for it. If you think you're not eligible, obviously if you're not a lady or a girl or a lady scientist, you're not eligible, then you share it with your friends who are female, and we would let them join us. We have only a max of 50 seats because there's just no way you can teach students if the numbers go too high. So I'm very happy that today we've not gone to the maximum number, but we are somewhere around 30, which is a very handleable number. But for this workshop, we have 50 seats and we intend to fill all the 50 seats. So you're welcome to share it in your circles, and you're welcome to attend it if you can. And you're welcome to tell your friends about it. Okay. I think this is time because I have another meeting which starts in 15 minutes. So I'm very happy to have been here today. I do hope some of you have learned something. In case you have more questions, please feel free to put them on the what's that other chat box we've got? The
Discord.
The Discord, yeah, let's get there. I'll be in the other meeting, but I will keep checking my messages in between, and I'll try to respond if I can. Thank you very much.